And welcome to the 10th Annual Invasive Species and Exotic Pest Workshop presented by the Columbia Gorge CWMA. I'm glad you could all make it here. We've got a lot of attendees, and I think that number will continue to rise. I hope everybody is staying warm and safe, and that everyone has their power restored in those areas that were affected. And happy National Invasive Species Awareness Week. We're about to finish out part one here, and seen a lot of great outreach and project spotlights over the last week, so great job. My name is Troy Abercrombie, and I am the coordinator for the Western Invasives Network. I sit on the steering committee of the CWMA, and I am the 2021 vice chair of the Oregon Invasive Species Council. Really excited to be here. Thanks for the opportunity. They've got a great program lined up for you all, and just really looking forward to hearing some of these presentations. I'm going to hand it over to Courtney Gattuso, coordinator of the CWMA, for some general housekeeping, and then we'll get right to it. Courtney?
Great. Thank you, Troy. So as Troy mentioned, I am Courtney Gattuso. I'm the coordinator of the Columbia Gorge Cooperative Weed Management Area. Thank you all for attending our 10th annual Invasive Species and Exotic Pest Workshop. So we typically hold this event in person, and this is the first time we have held it as a webinar conference. So it's our signature activity of the CWMA, and we're just very grateful to be able to continue to host it for all of you despite the physical distance we are experiencing these days. So I want to give a big thank you to our co-sponsor for today's event, co-sponsors. We have the Washington Invasive Species Council, Washington State University Extension, USDA's Animal and Plant Health Inspection Service, and Skamania County Noxious Weed Control Program. And just many more thanks to all of our speakers and organizers for tackling all of the recent hurdles with the snow and power outages to just help this event come together. So yeah, before we get started, just a few housekeeping items. Today's event will be recorded, and the recording will be available for attendees. All attendees will be muted to avoid disruptions, and webcams will be disabled. So you could show or hide your control panel with the little orange arrow. And for those of you who might be experiencing audio issues, you could change your speaker settings or call in on your phone in the audio drop-down menu within the control panel. So we will have time for some questions after each presentation. This platform doesn't have a chat function the way others do, so we'll be using the question box in the side control panel during each question segment. So you'll be able to type in your questions for each speaker during their presentations, and our CWMA co-chair, Lucas Nip of East Multnomah Soil and Water Conservation District, will be our question facilitator today. So he's going to go ahead and read your questions out loud to our speakers as they come in, and if we have any additional questions after the allotted time, or we aren't able to get to them all, we'll provide the speaker's email address after each presentation. So there's a lot of you here today, and we'll do our very best to answer all of your questions and comments as they come in. So just please keep them concise, and thank you for your patience in advance. So we do have recertification credits for our attendees today. So we have 2 recertification credits for our Oregon Department of Agriculture pesticide applicators, and one of those credits is a core credit. We'll also be offering 3 recertification credits for Washington State Department of Agriculture pesticide applicators, and no partial credits will be available. You must attend the entire day to receive credit. So have your license numbers handy, and when we prompt you, you'll need to type in a code word or your license number in the questions box. So you will not receive credit if you don't type in the code word or your license number, whichever one we tell you to type in, so just keep that in mind. And I also want to just give a quick update on behalf of the Columbia Gorge Cooperative Weed Management Area. So for those of you who aren't familiar with the CWMA, we're a collaborative network of organizations and agencies all working together to help fight noxious weeds in the Columbia River Gorge. And that spans over both Oregon and Washington. So the CWMA has gone fully virtual since COVID-19 hit, as many of you have as well, but we're still operating the best we can by having remote meetings and events like this one today. So last month we held an election for half of our steering committee members. And I'd like to just take a moment to introduce those folks in their new positions. So for our Washington representative, we have Emily Stevenson with Skamania County Noxious Weed Control Program. And we also have Angie Kimpo with the Portland Water Bureau as our Oregon representative. And we have as our at-large representatives, Christina Mead with the Mount Hood National Forest and Kate Conley with Columbia Land Trust. And the other half of our members include Troy Abercrombie, who's our MC for today, Lucas Knipp, our question facilitator, Sam Leininger, and Marty Hudson, who's going to be giving us a Kahoot quiz later on. And we do have one vacancy on our steering committee, and that's our Education and Outreach Committee Chair. So if you or someone you know would be a great fit for this role, let me know and I can give you more information about that. And lastly, we're always looking for new partners to join in our efforts. So if your organization would like to become a member of the CWMA, I'm more than happy to help get new folks involved in our group. So that's all I have for now. Let's dive into it. With that, I'm going to pass the virtual mic back to Troy.
Thank you, Courtney. I want to introduce our first speaker. His name is Dr. Dave Coyle. And Dave is an assistant professor in the Department of Forestry and Environmental Conservation at Clemson University. His extension program focuses on tree health and invasive species management in forest and managed landscapes across the Southeast. And his research program examines ecological impacts of and management strategies for invasive plants. Dave completed his BA in biology at Luther College, an MS in entomology and forestry at Iowa State, and a PhD in entomology at the University of Wisconsin. Dave was president of the North American Invasive Species Management Association. He's on the advisory committee for the South Carolina Exotic Plant Pest Council, and he's on the South Carolina Invasive Species Advisory Committee. You can find Dave's outreach work on Twitter at Dr. Dave Coyle or Instagram by the same name, where he regularly posts about invasive species and tree pests. Welcome, Dave. Thanks for being here.
Thank you, everyone, and thank you for putting up with that. So we are going to talk about bounties for invasives and planting of the natives. So again, thanks for having me. Good morning out there, everyone. Afternoon here. I hope everyone's made it through the weather well and fine and all of that good stuff. So let's dive in, shall we? So we'll talk about a bunch of stuff today centered around Bradford pear. I'll give you a lot of information that's grounded in some good science, but I also need you to know that I've been doing this for a while and I have some opinions that you will unfortunately, or maybe fortunately, be subjected to, because I think that helps set the stage for the story as well. So that said, just to set the stage, are all non-native plants bad? Because I've been called a non-native plant hater. That is not true, in my opinion, right? Because there's a number of native plants that we use, we eat them. You don't see potatoes growing wild and feral all over the place. You don't see orange groves popping up on the side of the road. These are non-native plants, right? Potatoes, oranges, corn, but they are not invasive. They're not just growing all over the place. In a lot of cases, especially when it comes to horticultural type plants, location is totally key. If anyone knows me, you know I'm not a fan of bamboo, but this happens to be a place near where we live, and there's about a 2-foot by 20-foot section of dirt along the side of that building. Bamboo's not going to go anywhere there, right? It's not going to get out. It's not going to go feral. It's fine right there, honestly. It's not really doing anything. So that, I don't have a problem with that. It's when it's planted, when some of these things are planted and then they have the opportunity to go feral. That's when it gets sketchy for me. So with that said, let's talk about calorie pear. Most folks know it as the Bradford pear. We're talking about Pyrus caloriana. Here it is, right? There's no denying that this is a pretty tree. There's very pretty flowers on this thing. But if you've been around these things, when they are in bloom, you know they also have a bit of a pungent odor. I think this is my favorite description. Smells like rotting fish and semen from Business Insider in 2013. It is a distinct odor if you've been around them. The reason these things are here in the first place all gets back to this fire blight. Okay, so this is a deadly disease, or a disease of pome fruits, of pears, causes that damage. It's actually affects the leaves and the fruit, and it's brought in by honeybees, believe it or not. So it's a pollinator spread type of thing. So, you know, back in the day, they had, back around the turn of the 19th century, there was a big issue with fire blight hindering the pear production. So they had to figure out something to do. Otherwise, that whole section was going to collapse the economy. So they knew there was this Pyrus caloriana over in Asia. It's native to Asia, pretty resistant to fire blight. And so it was brought over to the US to try to halt that disease. They brought a few, seemed like it worked, but we knew we needed some more specimens for our experiments. And so what they ended up doing was going to this guy. This is Frank Meyer, very well-known plant collector. He is so much well-known that there's actually an award given, the Frank Meyer Plant Genetics Award. And if you ever eaten a Meyer lemon, he is the guy that found and brought that Meyer lemon over. So he was a big deal. He was a very well-known, established plant collector. They sent him over to China to get some of these Pyrus caloriana specimens to bring back for that very reason, to use them to deal with the fire blight. And holy cow, it worked, right? So these collections of Pyrus caloriana helped save that pear industry. And what they ended up doing was grafting, right? So they would take a scion from edible pears, as usually your European pears, Pyrus communis, and they would graft that thing to the rootstock of a calorie pear. So then you would have, you know, the resistance conferred from the calorie up into the top edible pear. But then, so there's always a but then, right? So they had several outplantings up in Maryland at the U.S. Plant Introduction Station. And this fella, John Creech, was the horticulturalist there. And he was looking around and he noticed that some of these selections had some really nice horticultural value. They were disease resistant, drought resistant, pollution resistant. And one of them, holy cow, it was thornless. It was also pretty, and oh my goodness, it was absolutely unkillable, right? These are the sort of things you need for a street tree. So he named it Bradford after the station director, and this is where the Bradford pear came from, right? So you've got these scions that were all Bradford, and they were these thornless, pretty, tough to kill shoots, and then they just grafted those onto general Pyrus caloriana rootstock. So this is an important part, right? The Bradford pear is a grafted cultivar. All the scions, again, are those top Bradfords, and then you've got these rootstocks of Pyrus caloriana, but all a little bit of different genetics. So this was marketed as a street tree, one of the first big street trees commercially released in the early 60s. It was first noticed to escape cultivation. It's the word they used in the mid 60s, both in Arkansas and in Maryland. In the early 80s, once some of these first planted ones got to be about 25-ish years old, they started noticing the structural problems, right? But by that point, it was a very, very common, very well-established tree. And, you know, the ball was kind of rolling. There have been lots of new cultivars created to try to deal with some of those structural issues, Aristocrat, Cleveland Select, etc. So the kicker is, here's what they knew. And when I say they, I mean USDA, because back in the day, USDA kind of ran this program. Clearly, we know more now than we did then. But at the time, it was pretty well known that around 25, 30 years old, even 20 years old, you had branch structure issues. And this picture I'm showing you is not some sort of anomaly. This is common across the eastern US with any Bradford pear. If there's an ice storm, if there's a snowstorm, if there's a rainstorm, if you look at it funny, it tends to do this in response to whatever you do, whatever happens. Bradford was also marketed as sterile, right? So Bradford pear is a sterile cultivar. The reason being, again, the top of that Bradford is all the same genetics, and it can't make a viable seed if you try to basically mate it with itself. What they knew, though, is if you have pollen from any other Pyrus, you will make a viable seed. And so what that means is, let's say you've got a theoretical town and every street tree in there is a Bradford pear, just like this. That being the case, you're not going to have any seeds because there's no pollen that's not Bradford pollen. All it takes is one person, that one neighbor, right, to go ahead and put a different cultivar in there. Let's say they put a Chanticleer cultivar in there, or let's say the top of their Bradford breaks in a storm and a sprout from the root grows up. That is no longer Bradford. right? So that's different genetics. So all of a sudden, you've got different pollen intermixing, and all of a sudden, all of these things can produce viable fruit. So the USDA response, when somewhat called out on this, was basically throw their hands up. They just said, look, this is a massive, major horticultural moneymaker for us. It is what it is, right? Everything's got pros and cons. They were willing to just use these. It was just one of the cons. Unfortunately, because of that, we find ourselves in this particular predicament today, especially down here in the Southeast. This is an aerial picture from just up the road where I live in northern Georgia, and this house has a line of Bradford pear trees. And you can also see across the road, now you have this great big patch of calorie pear trees, wild calorie pear. How did that happen? Well, the fruits of a Bradford pear are about the size of a marble, and they're very tasty to birds. Birds do what they do. They eat, they fly, and they poop. And that's how these things get spread all the time. So what you're seeing in this photo is the results of birds that will sit in those Bradford pear trees, eat those viable fruits, then fly off over the field and take a dump as they're doing so. And boom, you've got a patch of wild calorie pear. And because of this, we now have wild calorie pear all over the place. Our old fields are pretty much covered with it. If you leave them, if you don't mow them every year, you're going to have calorie pear sprouting up like this. We see wild calorie pear along the roadsides. It's about that time of year. Everything's going to start blooming. They're about the first thing to bloom. We see it especially in a lot of unsold lots because there's lots of Bradford pears in towns, and birds are kind of always hanging around there. You see it in the lots. And from my perspective, being in forestry, one of the big concerns is that we now see this stuff growing in inside forests. I mean, this is our standard southern pines and lines where we have it. It's usually burned every few years. Rows of these single-age pine trees. Calorie pear is establishing underneath that canopy of mature pines. So, when we look at where is Bradford pear, calorie pear at this point, this is what, one of our maps, our Ed map shows us, and this is really incomplete, this particular map. I think the range where calorie pear actually exists is now closer to this, and that's the Ed map's future range, right? So we know it's all over the eastern US. It's planted in a lot of municipalities in the western US. The only place that's really kind of avoided it so far is that central area of the country. But who cares, right? So this is a very pretty plant. There's no one that's going to, a few people will deny that this is a very pretty plant, right? And so that's one of the things when we deal with people, the public anyway, it's really tough to get them to think anything ill of this plant sometimes because it is so beautiful. But it's full of thorns, right? So this is just one of the downsides to the whole calorie pear issue is the thorns. These things will puncture skin. They will puncture skin on people. They will puncture skin on animals and livestock. We've had horse farmers come and say their coal, their little foals got into a bunch of calorie pear and scraped up their face. We have seen these thorns can pop tires on various different implements, everything from a riding lawnmower to a pickup to a tractor. So these are serious thorns that we have here. Ecologically, it's a fascinating thing because at this point we have so much, the genetics are so different out there because there's been so much intermixing and interbreeding with the Bradfords and the wilds and back and forth and back and forth. You don't have a monoculture out there of calorie pear. You've got a very diverse population that flowers and blooms at different times. Just in this picture alone, all of those trees are calorie pear. All these trees you can see in the front are calorie pear. And if you look close, some of them have barely started breaking bud. Some are full-on white, full-on flower. And some of them are already greening up because the flowers are falling off and they're starting to leaf out already. So there's a tremendous amount of variation in any of these populations across the land. And in fact, we had a master's student of mine at the University of Tennessee looked at the gene flow and all the genetics of calorie pear, and it turns out that it has pretty much all the characteristics you would find in a highly invasive population. I know for some of you this is a duh type of thing, but But it was really interesting to see, we sampled from all over the eastern US, from here to Texas to Kansas, Minnesota, up into the Northeast. And it's like a poster child for an invasive species. There's this, and then when you actually get down there, these thickets grow so thick, they pretty much block out the sun. This is from underneath a calorie pear thicket, and then this is the picture where I looked just down at the ground from where I was standing, and it gets so dark in there that nothing will grow underneath there except more calorie pears, unfortunately, that happen to fall down. So from an ecological perspective, it creates a dead zone in more ways than one, because it's going to crowd out all this other vegetation you have there, and it also does not provide a lot of food for caterpillars and insects that other things might eat, and we'll talk about that in just a minute. So at this point, over here in the east, we are looking at what to do now, right? So at one point, we need to figure out how to manage or kill it, right? In the southeastern US, we like to burn stuff. Prescribed fires in our forests are a big thing. Can you kill calorie pear with fire? Unfortunately, that is a hard no, because there have been studies that have done that shows if you burn a calorie pear, if you've got that old field, you send a prescribed fire across there, you'll top kill it, but then for every stem that you kill, 4 tend to come up, so it comes back angry and mad. So that's just not a viable option unless you pair that with something else, mechanical or chemical. Can you use biocontrol? I alluded to it earlier that it's not going to work at all, right? It's just that there's way too many, nothing eats it, right? We've tried that, we've done observations, we've done the laboratory work. There's a lot of generalist things, especially, that would rather not eat and die than eat a calorie pear. So biocontrol is simply not an option either. So what about just straight cutting it down, right? Yes, that will work, but that's only half of the equation, right? You can cut it down. If you simply cut it down and don't treat that thing with an herbicide, treat that stump, you will be, the treat you will get is a whole field of calorie pear coming up in your yard or in the field, wherever you take that. These things are prolific root sprouters and stump sprouters, especially if cut down. It's not unheard of to cut one tree down that was in someone's yard and get upwards of 50 sprouts coming off all the large roots that fan out there. So we know herbicides work, so we looked at that a little bit the last couple of years. We did some herbicides. This is one of our field sites right here, trying to figure out, at this point, we're trying to figure out what can we do for land managers who've got this in their fields. You can't mow this over when they get this high. These trees are 6 to 8 feet high. There's thorns all over them. You've either got to, get like a big chipper on a track out there. It's just a lot of effort. So we're trying some herbicides. The good news is they pretty much all work, right? Glyphosate, hexazinone, imazovir, triclopyr, all of these things work well. We had one issue with one of our treatments in Bartram because the rainfall was funky. It was too dry when it was applied, and it was too wet when we were trying to measure. But by and large, all these things work, right? So herbicide works fine. The kicker here is that you've got a lot of people that want nothing to do with herbicides. They think that herbicides are the devil, when in actuality, if you apply these things by the label in the right way, they are not the devil, right? They're an excellent tool in our toolkit. So because of that, we tried to think of, well, what can we do that's going to be a little bit different? What can we do to engage people, and what can we do to try to, as I said in my title, steer people the right way? And 3 years ago, there was a program that happened in the city of Fayetteville, Arkansas, where they were asking residents to chop down their Bradford pear trees. And in doing so, they would give them a free native replacement tree. I believe that same year, the invasive species group in Missouri had a similar program. Seemed to go pretty well. I spoke with some of those folks, and they had a fairly good experience there. And I thought, well, maybe this is something we can try over here. Maybe it's a model that can be made. And I think it's important because a lot of these invasive plants, especially these invasive landscape plants, are easily obtainable and sometimes promoted. For instance, Japanese bloodgrass, otherwise known as Imperatus cylindrica, or Kogan grass, which is a major invasive weed down here in the southeast. You can buy cultivars of Kogan grass that are promoted like this as a as an ornamental. But I tell you, the minute you take and plant them in a warmer climate, they just turn green again and go feral back into kogon grass. We've done that. So this does not hold. It may hold in a pot or something, but not when if it happens to get out. We've got, the National Gardening Association that is promoting wisterias and that type of thing. And if you've ever been to the Southeast, there's Chinese and Japanese wisteria. These things are prevalent all over the place, and they climb up mature trees and just weigh things down. It's a really horrible invasive weed. And to that end, I can go to Lowe's up the road and I can get a flowering Pyrus caloriana today if I want to, for the low price of $26.98. You still have these plants which we know are not good for a whole bunch of reasons still being sold. And honestly, if I don't want to go to Lowe's, I can sit here in my office and I can buy one at social distance. All you have to do is Google purchase Pyrus caloriana, and all sorts of options pop up for you at the click of a button. Some of these as low as like 20 bucks or so, right? So there's a big problem because a lot of these plants that we know are invasive are still very much available for horticulturalists and landscapers and anyone to just go by and put in there. And I would like to think at this point, a lot of the professionals, a lot of you folks that are out here watching are probably aware of what, which of these things are good and which are not, but there's a lot of landowners that don't have a clue. There's a lot of landowners that see that pretty white flowering tree in Lowe's and think, I want that. There's a lot of landowners that look down the street and see their neighbor's got that fluffy looking white flowery tree in the spring and they say, I want that, and then they would go buy that. So in coming up with all this and realizing that, we are never going to get rid of all the calorie pear that's out there, that's just simply not going to happen, but what can we do to raise some awareness of it? And one of the great things about Pyrus caloriana is it shows off every spring. It's going to start here anytime it flowers. It's about the first thing to flower. It turns the roadsides white. It turns the fields white. It's just beautiful white flowers everywhere, and everybody notices. It also has that odor, which everybody also notices, for better or worse. So it's a very noticeable tree. It shows off. It's showy. So we decided to capitalize on what had already been done in Fayetteville and Missouri, and we started Clemson's Bradford Pear Bounty Program. We did this last year, and we had a pretty good time with it. So we're going to talk about this for just a minute. So first of all, the who does the Clemson Bradford Pear Bounty? Well, it's obviously a cooperation between Clemson Extension and then our South Carolina Forestry Commission. So our state forestry agency has an urban forestry people in there. And then we partner with local municipalities, right? We did it last year with the city of Clemson for obvious reasons. We're right there. This year we have added another city, Sumter, South Carolina. So Clemson is way up, if you're not familiar with our geography. Clemson is way up on the western corner of the state. Sumter is much more to the middle of the state. So we're in a very different geographic location. So then what are the roles of all these different entities in there? We've kind of figured out how things work at this point. Clemson Extension is more or less responsible for all of the infrastructure, the electronic infrastructure, and the PR. Okay, so we have our communications team puts together the things that can be shared on social media. They put together, all the website, which we'll talk about. And then we also handle the media. And when I say we, I mean me, basically. This generates a considerable amount of news coverage, both TV, radio, there's been podcasts, there's a lot of newspaper, there's press releases that go out. This year it was the number one, the number one story on one of the Columbia, South Carolina news stations for 2-3 weeks in a row is the Bradford Pear program in Sumter. Sumter's not too far from Columbia. And every day or every other day, I was having media requests from them to want to talk about it. Just today, how many people have registered? Are you going to go set up tomorrow, et cetera, et cetera. So it generates a lot of response because a lot of people know what these things are. And there's actually a pretty big hatred of them. Even though there's a lot of people that will still buy them, there's a lot of people that want nothing to do with these trees. at this point anymore. So that bodes well for us, honestly. And then also there's the website, and this website took a considerable amount of time to develop. I've just shown you the different sections we have in there, right? So we've got some basic things about the Bradford pear bounty, and this is just if they click on that, what it is, locations, times, registration links, basic bare bones logistical facts. And then there's sections about flowering pears, right? The difference between Bradford, calorie, the register. We talk about what is invasive, because after all, we are an extension organization. We want to educate people and talk to them about, you're not just getting a free tree because we think you're cool, even though they are cool, but here's why we're doing this, right? We want to teach them something while they're there, and then help them recognize that all those white trees they see out in the landscape are there because people have Bradfords in their yard, right? We've got some of the safety stuff, how to properly remove the tree, because that is part of the deal. We ask people to cut their tree down and take a selfie with it, and then bring that selfie with them, just on their phone is fine, and show us at the event, check their name off, and then they go get their tree. Lots of information about tree planting and tree care. A lot of folks are very clueless on how to do this. And then we have a whole section on the replacement trees. Now these differ by location. Clemson is kind of up in the mountains, and then Sumter is much more coastal plains. So we've got some different species. Not everything works everywhere. We talk about sponsors. In some cases, this year we've got a corporate sponsor that purchased the trees for Sumter. And then we've just got, a form where they can write in if they have questions, some of the FAQs, the standard stuff, and then we encourage people to share their bounty photos when they're done. So this is Clemson Extension's side. The Forestry Commission, we ask them, they help provide some of the trees. The way it works is they've got some funding and support to purchase trees for public places. They help set everything up. They're involved from day one, and then they help do some of the promotion as well. And then from the actual municipality itself, we ask them to handle the logistics. So the trees come in a couple weeks early. We ask them to take the trees, put them somewhere safe, water them if they need it, and then get them back to wherever we're going to do our program. That morning, it's generally at a park, at a pavilion, that type of thing.
Hey, Dave.
This is how we've done it so far. It seems to have been working out well so far. But it's a little bit different. We tried to mesh what we're going to do and what we're trying to do with each municipality. So
hey, Dave, got a 5-minute warning. Can you hear me? 5-minute warning?
You got it. Thank you. So we're at the recap already. When it comes to calorie pear itself, management is difficult, but it's possible. But it's going to cost money. You're going to need to use herbicides or some other type of expensive equipment to get it out of there. Clearly, with any invasive, it is way better to prevent these things from getting out than to have them. But at this point, we've got this Bradford pear bounty. It's been very successful in educating people and raising awareness. We've got over 200 people registered for each event so far, and this year alone. So we feel quite good about the trajectory of where that's going, especially with the considerable press that it generates. And with that, I just want to thank all my collaborators. We've worked with GFC and SCFC with the research, and the Forest Service, and our cities that have worked with us, and then Shawani's up at the University of Tennessee as well. And with that, I have time for questions, and if anyone would like to know more, I would be happy to answer, happy to email. But yeah, happy to take any questions at this point about this type of program.
All right, Dave, we got a couple questions here in the chat box.
Sure.
Someone asks, is Nandina domestica Moon Bay truly a safe, non-invasive fence border plant?
No, we're seeing Nandina get out in the natural areas already. So I do not believe Nandina is going to stay where you put it. To me, that's one of the sort of emerging invasive plants that we're going to have to deal with, because we're seeing it, and I've got colleagues down here that we're seeing it out in the wilderness already. So I don't think Nandina is going to stay put personally.
Good, thank you. Someone else asked, is the Bradford tree on any state quarantine list? And could that help with the distribution problem?
Yeah, it's on a few. I think Ohio put it on there. We are working to get it on South Carolina. That's a tricky one because it's got a lot of industry backing. It's a Pyrus caloriana is a rootstock commonly used for fruit tree grafting, right? So it's a very common rootstock. So when you say you want to ban Bradford pears, then all of a sudden, well, do you mean just the Bradfords, or are you going to ban the rootstocks too? That's a separate animal. It's proven much more difficult than we thought it would. I know several other states are working on it, but because of that tie to the fruit industry, it's complicated, I guess is maybe the best way I can put it.
All right. I don't see any other questions in the chat box. Speak now or forever hold your peace with Dave. Oh, we got one.
Oh,
That worked with. You ready?
Yeah, here's one from Sam. What was the total cost of the Bradford pear bounty program? That's what I see popped up here.
Okay.
And I can handle that. We have a tree nursery we deal with in the Panhandle that has the right climate to grow stuff for us. It ends up being roughly $10 a tree when you factor in cost and delivery. Now each species is a little different, but roughly we got 250 trees for each event, roughly $2,500. The rest of it is all just time, right? It was my time to create that stuff. It's my time to, our College of Ag communications time to do the website, and the municipality's time to take care of trees. Other than the tree cost, you're looking at, a couple $100 because we'll get, well, I don't think we will this year, but coffee and donuts in the morning for people to come and attend, just something like that. So it really isn't a huge cost, provided you can get trees at a reasonable price. Other than that, it's mostly just time.
Great. And we got one more question. We got a couple more. I think we'll probably just go with one here. First one that came in: with the bounty program, the landowner does the control. Do they then quickly realize the value of herbicides?
We hope so. I mean, that's the goal. We give them a lot of information about when you cut that tree down, you've got to treat that stump. And we've had plenty of people that didn't treat the stump. And then, of course, we get emails, 2 months later, why do I have 50 Bradford pear seedlings in my lawn? Well, did you treat the stump? Well, no, I don't like pesticides. Well, there you go. It's one of those deals. You can't, can lead a horse to water, right? But you can't make him drink. So we do what we can, and people either figure it out or don't.
All right. Yeah. Well, I've been informed that we have more time for questions. I was incorrect in my time, so I want to keep asking you questions because they're pouring in.
Yeah, sure.
The next one asked, what natives did you replace the Bradford with? I think is they're asking, or what options are there for native replacements?
Yeah, there's quite a few. At Sumter, which is again more coastal plain, so it's deep sandy soil, a little more humid climate down there. We've got a number of different oaks, pawpaw tree, a number of flowering bushes, viburnum. American beech tends to grow well, bald cypress grows well, black gum grows well, Nyssa sylvatica. Up in Clemson, it's a lot of the same, but we don't do some of the stuff like a pawpaw, or we don't have all the oaks. There's a white oak, swamp chestnut oak, boy, I can't even think, overcup oak. There's a whole bunch. There's about 12 to 14 different tree species we have. So it varies a little bit depending on where you are, and that also changes year to year a little bit depending on what the nursery has in stock.
Cool. I don't know any of those species, but you sound like you know what you're talking about. So if I was a landowner, I trust you. I think I just need one more here. If any more come in, we still have time, everyone. Someone asked, could you compare the invasiveness of the Bradford pear to other invasive fruits? And in parentheses, they have cherry, stone fruit.
I think the Pyrus caloriana is on the high end of invasiveness just because of how quickly it grows and how rapidly it can take over places. I have not seen a whole lot that parallels it, other than things like privet. I put calorie pear right up there with privet and honeysuckle in its ability to quickly colonize. It's tough to kill. If you cut one down and don't treat the stump, it's going to send up a boatload of sprouts almost immediately. it's super aggressive. It's super aggressive. And really, if you've got an old field down here and you don't mow it for, by the second year, you're probably not going to be able to mow it because there's going to be too much calorie repair and the stem's going to be too woody to even use a bush hog with. So very, very invasive. And I wanted to make a quick point, Lucas, on what you just said, about knowing what to replace a cut down Bradford pear with. One of the things we do at that event is we have me there and some of our clumps and extension folks. We've got the Forestry Commission folks there. We've got the local city horticulturalist or arborist there. And a big part of what we do that whole morning is just talk to people about what do they want. Because most people that come to this program, they hate their Bradford. They have no idea what else should go there, right? And so we help guide them and help them choose the tree. If they had a Bradford pear under the power line and they come in wanting a white oak, we're not gonna let that happen. That's a horrible idea, right? So we'll steer them towards a smaller tree or one of those flowering shrubs. But if they've got it where they can plant it in a wide open space, well, yeah, if you want something for shade, an oak is a great choice right there. So that is one of the good things we have. And again, that's just our time, but we take the time and advise people because very few people come in knowing exactly what they want, unless it's a dogwood or something, they want a flowering ones, those always go first. But it's a very interactive process where we help people figure out what they need.
Cool, sounds great. I'm gonna try to blast through these last questions. As we're running out of time, maybe one or two. Someone just commented, great information on invasive fruits. I guess more of a comment than a question. They said we have a lot of escape plums in Western Washington. So they're just saying thanks for the info. Someone asked, are the replacement trees bare root?
No, we get them in 3-gallon pots. So they range from 2 to 5 feet tall. And we chose that because we felt like it'd be harder to kill because we know a lot of these people probably aren't that experienced in tree planting, so they're harder to kill. And also people, I think they feel a lot better. They take home a legitimate tree in a good sized pot, right? It's not like you're giving them a little dinky seedling that their kid's gonna mow over when they mow the lawn in a month. They're getting an actual tree, and I think people seemed really happy and very surprised that, oh wow, this is a real tree. So that was one of the reasons we went with something a little bit bigger than a seedling. I know it makes it a little more expensive, but we just felt like that might improve the overall survival potential for it.
Great. We have some more questions, but we're going to kick those over to you in your email, I think is the method that Courtney is going to employ. So we're out of time. Great talk.
Thanks
for answering all the questions. I'm going to kick it over to Courtney.
All right, thanks.
Great, thank you so much, Dave. So now we're gonna take a quick break, 10-minute break. So we're gonna come back here at 10:55. So we'll see you guys soon.
All right, welcome back, everyone. Clock on the wall says 10:55. Great presentation by Dave. That was really awesome. And our next presentation is going to be about Asian giant hornets. And our next presenter is Cassie Cichorz from the Washington State Department of Agriculture. Cassie is the Asian giant hornet outreach and education specialist at the WSDA, and her goal is to prevent this invasive pest from establishing by helping deliver project information, implementing outreach activities, and working with citizen scientists. Thanks for being here, Cassie. It's all you.
All right, I just want to make sure you can hear me. Okay, perfect. All right, well, thank you guys for having me. Like said, I'm going to talk about Asian giant hornet. And let me put my slides up for you. Basically, I'm just going to give a brief overview about Asian giant hornet, talk about what Washington State Department of Agriculture is doing, and at the end, I'm going to go over a couple of our native species that may or may not resemble Asian giant hornet to you, just so you can see the differences and get a little bit more on that pest management in. So Asian giant hornet, Vespa mandarinia. They're also known as yak killer hornet, giant sparrow bee, and I'm sure you've all heard the infamous murder hornet. When you're talking about them, Googling them, I ask you to use Asian giant hornet instead of murder hornet, not just because it's its name, but it's going to give you the most correct facts when you're looking things up. Basically, murder hornet became sensationalized by the media. When Asian giant hornet was discovered in Washington state in 2019, an article was published by the New York Times titled "Asian Giant Hornet Threatens Honeybees in the Pacific Northwest." It gained a little ground. Following that, they interviewed some researchers overseas, followed the WSCA, as we said, experimental traps, then published that article with the word in it. So just use Asian giant hornet, give you the most bang for your buck. So what are they? They are the largest hornet in the world. Adults range between 1.5 inches to 2 inches in length. You can tell them apart by that distinct large orange-yellow head and the orange-yellow band striping on their abdomen. They are native to Asia, most observed in the areas of Japan and Korea. They like subtropic to moderate temperatures, hence their attempt to establish in the Pacific Northwest and Washington states. So I always hear people argue they're definitely orange or they're definitely yellow. I'm always going to say they're orange-yellow. Think about a school bus. I'm going to say school bus is yellow, but if I'm shown a paint swatch, I'm going to lean more towards orange. So when did we discover Asian giant hornets and why are they a problem? We found Asian giant hornets. A resident from Blaine reported a specimen to the Washington Invasive Species Council in December 2019. Then WSCA confirmed it to be. Asian giant hornets are not native to here, and they specifically threaten honeybees, can harm human health and agriculture in Washington state. Any invasive species has the potential to damage any habitat, climate, destroy a niche, take a food source away, but as I go throughout this PowerPoint, the specific threats to honeybees, human health, and agriculture will become more real for you. So a couple things I want you to take away from this is being able to identify and know what to do if you think you see an Asian giant hornet, but also a little bit information to understand why the WSDA traps, how citizens can help, and more information for you to share with others. So a couple things about Asian giant hornet. Typically non-aggressive, but if they feel threatened, they can sting. A couple times that they might feel threatened is if you come upon a nest because they want to protect their home. Asian giant hornets actually typically nest in the ground. What they'll do is they'll take over at the base of a pine tree or in an old animal or rodent burrow, and they'll build these underground nests that can be over 2 feet wide and produce hundreds of adult hornets. In their native range, there is occasionally a nest recorded in a tree cavity in an above ground or in a hollow tree, but typically these nests are in the ground. Since they're hornets, they can sting multiple times. So if you come upon an Asian giant hornet nest, you might not see it right away because it's in the ground, and this is a chance you could become stung or someone could become stung. So here's a video of a lone Asian giant hornet hovering above a nest. So actually in September of 2019, a nest was found outside of Forest Park, pretty much directly off a path that people were walking in Nanaimo, British Columbia. This nest was removed and eradicated. So the other really important thing to take away is the life cycle of an Asian giant hornet. This will help you understand the species better and understand trapping that the WSDA does. So I'm going to spend a minute on this graphic. I'm going to start at the top and go in a clockwise position. So in the winter, the males and the workers and the queen that started that nest, they die. The only thing that will make it through the winter is new queens. What they'll do is they'll find a sheltered spot or a spot in the soil, and they'll do what we call overwintering. Moving into the earlier spring and into the spring, these queens will stop their overwintering. They'll emerge and find a spot to build that usual underground nest. Moving later into the spring, into the summer, you'll see workers out. They'll be flying around, foraging, finding foods to bring back to their colony. Asian giant hornets are an apex predator, so they eat carbohydrates, but they're also capable of eating other insects. So these workers throughout the summer will be out flying around, bringing food back to their colony. Moving later into the summer, early into the fall, that colony will reach its maturity. So that queen is going to stop laying workers, and she's going to start laying new males and virgin queens. So moving into that fall part where those new queens and the males are being laid, there's a potential need in their diet for more protein. And what is a better source of protein in their native range than honeybees? So this is the chance where because of that fall time, since the new queens and males are being laid, they might need more protein in their diet. This is where honeybees become what we like to call the danger zone, where Asian giant hornets might decimate an entire hive of honeybees to fill that need for food. So that's that fall time. Once the queens and the males are laid, then they will emerge from the nest. They will mate really close to the nest, and then, the queens will fly off to overwinter. The males and the workers will die. A couple things, unfortunately, the dispersal distance of queens is kind of unknown. There really isn't a lot of research to say how far queens will fly, or if after they overwinter, they'll fly off again. So that's something that makes it a little difficult in trying to identify, where the nests are and how far a nest could be the potential next year. I'll talk about how we do our trapping a little bit later on and distances with that. But going back to that fall time, right? I talked about honeybees being in danger. I want to talk about it a little bit more. So if Asian giant hornets have that need and they find a honeybee hive and they decide it's a great source of protein, here's kind of what happens. A couple Asian giant hornets will kind of start hunting around the hive, maybe picking off an adult honeybee or two, mashing it up into a ball and taking it back to feed their hive, or their colony. If they decide this honeybee hive is a great source of protein, they will mark it with a pheromone that will signal the other Asian giant hornets to come here. Following that, they will begin to enter the hive into what we've heard called the slaughter phase. What happens during this phase, they enter the hive, grab the adult honeybees, decapitate them, and drop their bodies to the ground. Once the adult honeybees are taken care of, they will start to occupy the hive. They'll defend this hive as their own during this phase because they're taking the brood and the pupae from the honeybees back to their own colony to feed their own. They're going to occupy this hive until the brood and the pupae is no longer viable or they don't need that food source anymore. Normal beekeeping attire doesn't keep people safe from the sting of an Asian giant hornet. And because they're occupying this hive as their own, if someone comes upon this while the Asian giant hornets are decimating a hive, they could become stung. In their native range, a couple of facts and figures they give is 1 to 20 Asian giant hornets can decimate a hive anywhere between 1 to 6 hours, and on average, it takes about 90 minutes. So I mentioned a normal beekeeping suit won't keep you safe from the sting of an Asian giant hornet. Since they're a very large creature and they're a hornet, they can sting multiple times, and their stinger is capable of delivering more venom than we're used to if we're stung by that of our native insects. So just really quick, what people should do if they believe they've been stung by an Asian giant hornet, they need to apply ice, take an antihistamine. If they're in pain, take aspirin and then clean it. If people are allergic to bees, wasps, or hornets, they're likely to have an allergic reaction, so epinephrine and call 911. However, that hornet, it can sting multiple times, and if stung multiple times, this is when it becomes more serious. So applying ice, antihistamine, taking aspirin if you're in pain, and then seeking a healthcare provider promptly is what people need to do. If they're allergic, they might be likely to have a severe reaction, so epinephrine and call 911. In their native range, Asian giant hornets tend to kill 10 to 20 people each year. However, there has been years where deaths have been reported up to be close to 40 people. So it can become serious on human health and honeybees if Asian giant hornet were to establish. All right, so if these are so harmful, why would they still want to have them in Asia? Well, other various reasons, but what happened was in the past, protein was definitely needed for humans to survive, right? And Asian giant hornet became part of a diet as a good source of protein, a good viable food source. It kind of cultivated to this day to actually become now a delicacy. The pupae and the larvae from the Asian giant hornet nest is actually harvested and then, kind of maybe added some seasoning, fried up, and it turns into a feast. I said delicacy, so kind of equate it to be about $31 a pound here in the US. Also, they will eat adult Asian giant hornets. They'll fry them up. Sometimes they get put into energy drinks as a novelty. Kind of like we've heard us over here, people do the tequila and the worm, maybe the hornet and the alcohol is another thing. So that's just kind of a little bit of background on the Asian giant hornet overseas. All right, let's flip it back here to Washington though. Specifically Whatcom County. Where have Asian giant hornets been found? Only in Washington state, other than in British Columbia, specifically in Whatcom County. This map on the left is a picture of Washington. All the orange dots are submissions people have reported to the Washington State Department of Agriculture as potential sightings. Those orange dots are unconfirmed sightings. The red circles and the red squares are confirmed Asian giant hornets. In 2020, there was 31 confirmed sightings of Asian giant hornets. Half of those reports came from the public, so it's awesome, and we give a big thank you to the public. To detect and detect the spread of Asian giant hornets, it's really crucial for us to trap, but also to keep the public involved in reporting. I might always get asked what's going on in British Columbia, so I'm just going to tell you that last year there were 6 confirmed sightings in British Columbia from the area of White Rock to Aldergrove. So in Nanaimo, where the nest was eradicated, across the waters last year, no more Asian giant hornets were confirmed. They were just confirmed across the border. So what does it look like when we have a confirmed sighting of an Asian giant hornet? What do? Well, we create what we call response grids, and this tells us where we're going to trap or where we're going to survey. Take one of those red circles, and that's where an Asian giant hornet was detected. From that, we're going to go out 8 kilometers and create a circumference around it. I say 8 kilometers because that's the most usual foraging distance, how far Asian giant hornets are going to fly from their nest to find food and fly back. They typically won't fly farther than that, so we're going to survey out 8 kilometers from any detection we have. This map you see here is what we will be surveying this year in 2020, starting in July. So there's that area of Blaine, Birch Bay up to the top left, and then in May of 2020, one deceased queen Asian giant hornet was detected in Bellingham. That's the additional square or circle on the bottom half, so we'll be surveying there as well. You might see that portion though off to the right-hand side with the area of kind of Linden. No, there's been no confirmed sightings of Asian giant hornets there. However, across the border in Canada, there was Asian giant hornets confirmed, so we want to survey in that area as well to make sure we're not missing anything. That's in addition to this year's survey that we didn't have last year. Last year, Washington State Department of Agriculture placed over 900 Asian giant hornet bottle traps. However, it's not just this Washington State Department of Agriculture that's trapping. We're reaching out to citizens in Washington and asking them to help us detect and limit the spread. Last year, citizens responded immensely, and we can't thank them enough for helping us. Citizens and cooperators deployed over 1,500 Asian giant hornet bottle traps in the state of Washington. So they took their time and their resources to check these traps each week. What these traps look like, we use the same ones. It is a 64-ounce, 2-liter clear plastic bottle. We make the bait with rice cooking wine and orange juice. You hang that bait 6 to 9 feet up in a tree, and then you check it each week. What you do is you strain the contents through a strainer, look at the insects, and then you report your results back to Washington State Department of Agriculture. Right? This year, we will be asking for citizens to help us trap and survey again. So if you're in the state of Washington and want to get your hands on being a scientist and help predict the spread of this invasive species, look at our website in July and we'll have those details out for you. So we have these traps, right? The Asian giant hornet's going to fly in, it's going to drown in that rice cooking wine. It won't be able to fly out, and we'll have a deceased Asian giant hornet. However, our goal is to capture a live Asian giant hornet. The reason we want to capture a live Asian giant hornet is because we want to find the nest. Typically, by nature, the worker will forage and fly back to its nest. So if we can capture that live Asian giant hornet, we can hopefully find that nest and then remove that nest. If we remove that entire nest, we're removing that entire population and not allowing it to go forth into the next year. So what we do is we modify an Asian giant hornet bottle trap. The Asian giant hornet will fly in, it won't be able to fly out, and then we'll have one of those live ones. What we do next is we attach a tracker to the Asian giant hornet and then, follow it back to its nest. A lot easier said than done. What we use is we use the process of telemetry. So basically, our entomologists are going to be walking around the forest with what it looks like to be an old cable and TV antenna. As you are pointing this antenna in the right direction of the tag or the transmitter, which is on the Asian giant hornet, you'll hear a feedback. So it almost sounds like a click. And as you get closer, you're in the right direction, that click becomes stronger and louder, so you know you're in the right direction of the Asian giant hornet. These nests are typically in the ground, so it might not be the easiest to see. So another thing we have equipped with us is a flare. That's the picture in the bottom right hand. If you point it at the ground or at a tree, and there's a heat source behind it, that will light up and help us confirm that the Asian giant hornet is there, as well as the visuals of seeing it enter and exit the nest. So in October 2020, last year, a couple of months ago, The first nest was found in the United States, and then it was eradicated. Once we find that nest, we want to remove that nest. That way that population can't produce new queens. They can't fly off to make new nests the next year. So what we're going to do is we are going to kind of douse or sedate the live Asian giant hornets that are around the nest. So we're going to douse them in carbon dioxide. We're going to capture the live Asian giant hornets, and then we will begin to remove that nest. The nests we plan for were typically in the ground, so we remove that entire nest and take it away. However, sometimes they might be in a tree cavity, so then we will have to seal that entrance off, remove the adult live Asian giant hornets, wrap that tree off, and then remove the tree. I wanted to show you a couple pictures of what it looked like when we got into that nest. So what we did was we cut that tree cavity open, removed the adult live Asian giant hornets, and then began to examine the nest. A couple things we found. I want to show you this picture on the bottom left-hand corner. That's one of the combs. So that's what the Asian giant hornets hatch out of. Those white marshmallow-looking things are a capped cell that has an live or Asian giant hornet underneath it that hasn't hatched yet. And the other cells that are empty have hatched, or you can see on one of the pictures it has a piece of larvae in it. So when Asian giant hornets hatch, they don't look like baby hornets. They don't look like little animals. They look like an adult Asian giant hornet once they hatch and leave the nest. You can see in that the orangeness in that picture there, that's an Asian giant hornet that was getting ready to hatch and emerge and leave. All right, so this is my last talking point before I run into some common imposters or insects that might look like Asian giant hornet to help you detect them a little bit better. Washington State University and some researchers did a climatic study to see what the potential spread of Asian giant hornets could be. They did this for more places than the United States, but I'm just going to highlight this for us. Looking at this map, the red to yellow pictures are just climate that Asian giant hornet could potentially survive in. So it's not looking at habitat, food sources, really basically just climate. So as you can see, that red yellow in the Pacific Northwest looks like pretty good suitable climate. And then you see kind of a blue-purple breakup as you move across the Midwest, the United States. Climate there isn't very suitable for Asian giant hornets to survive in. And then as you move closer to the East Coast, you see they have a potential there again. So this helps predict the potential spread of Asian giant hornets. Saying Asian giant hornets on their own really couldn't go from the East Coast to the West Coast. They wouldn't be able to fly themselves across. They would have to potentially hitchhike or find a ride to get them across. And as we know with any invasive species are good hitchhikers and they like to spread. So our goal here, is to eradicate Asian giant hornets from Washington State to protect human health, honeybees, and agriculture, but also to not let them spread across any of the Pacific Northwest or any potential habitat they might encounter. All right, last part I'm going to have for you. I want to leave some time at the end for questions. So I'm running about 18 minutes now. This is the size comparison of an Asian giant hornet and a honeybee side by side. Obviously, we want to keep our honeybees. Same with our bumblebees, small, black, yellow, and fuzzy. Fuzziness, black and yellow, not an Asian giant hornet. This is a bald-faced hornet compared to an Asian giant hornet. They're found throughout North America, pretty common in the southeastern United States, actually. Usually about a 1/2 inch to 3/4 of an inch, a little bit bigger than yellow jackets. You can identify these because they're mostly black, and then their abdomen has white markings on them. Great golden digger.
Hey, Cassie, we got a 5-minute warning. Yep. 5-minute warning. Thank you.
Right. Let me roll through this. Great golden digger wasps get confused as well. They are brown they have orange legs, unlike the Asian giant hornet. Their abdomen is orange with a black tip. They're missing that banding of Asian giant hornet. Cicada killers found throughout western United States, about 2 inches long. Their head is much smaller in proportion than that to an Asian giant hornet. So if you look at that head of that Asian giant hornet, it's pretty big, pretty wide in comparison to its body. Their banding is different. Last thing, the thorax tends to be more red, whereas the Asian giant hornet in the middle tends to be more brown. Horntail wasps get reported a lot. If you look at that, that looks like a scary thing at the end of it, a very big stinger. Well, you can see it's not a stinger compared to the Asian giant hornet. It's actually an egg-laying tube, and this is where they get their nickname wood wasp, because they lay their eggs in wood. A couple more for you. These are yellow jackets, found throughout pretty much all the United States, different where you find them. About a 1/2 inch long, but sometimes queens can be larger. They're pretty much brightly colored, and they have different markings than Asian giant hornet. Sawflies get reported as well. found throughout North America, 3/4 to an inch long. They're missing a wasp waist between that thorax and the abdomen, unlike Asian giant hornet. It tends to be more of a white or yellow spot. Fun fact, largest sawfly on the continent. Last one here for you is paper wasps. They can get about 3/4 an inch long. They usually have a very well distinct defined wasp waist to separate them from other hornets. All right, the thing you need to do, or anyone needs to do if they think they see an Asian giant hornet, is report it to the Washington State Department of Agriculture. Any details of what they saw, where they saw, what you noticed the hornet was doing needs to go with it. If you or someone can snap a photo, that's even better. Without a picture, it's hard to confirm or deny if it is Asian giant hornet. If the specimen happens to be dead, go ahead and collect it in case we want to test it later. Where to report it? Our website has an online reporting form, agr.wa.gov/hornets. Shoot an email to hornets@agr.wa.gov, and then we do have a hotline where you can phone in. If you're not reporting but you want to keep up on what the WSDA is doing, connect with others about the Asian giant hornet world, we have a Facebook group we run and monitor, www.facebook.com/groups/hornets. Here's my email for you. If you have any questions, go ahead and send them my way. Sorry I didn't leave much time for questions. I cut out a lot, but if there's any questions right now, I'll take them.
Thanks, Cassie. We actually do have time for questions. I see one first off from Troy that says and he's referring to your map where there was kind of a gap. And he said, when you see that big gap to Bellingham, do you make any assumptions on the reports in the space between, or that it was transported to Bellingham from elsewhere?
Well, the distance of the dispersal distance of queens is unknown. So it's unknown if the queen was able to fly herself down there or if she hitchhiked a ride. But the foraging distance is 8 kilometers, so there's no reason from the Bellingham specimen, we went out 8 kilometers. From any other specimens, we've gone out 8 kilometers. Unfortunately, there's that gap where there's no 8-kilometer distance from any other detection for us to trap in.
Okay. Okay, a lot of questions coming in now. I have heard reports of AGH in Oregon. I am guessing these are not confirmed. Is that correct?
Correct. And if anybody has a report in Oregon, they need to contact the Oregon Department of Agriculture to report that.
Cool. Someone asked about the traps. Are there any other species that commonly show up, and could these traps harm native species?
So they're deterrent to our native beneficial pollinators. They will trap other hornets like bald-faced hornets and potentially yellow jackets.
Okay. Since the species is native in a subtropical habitat, could the cold winters, hard freezes help decrease survival in North America?
Actually, in their native range in Asia, they do have some pretty harsh winters there. So there is no really rhyme or reason saying that the winter here would keep them from establishing or moving forth.
All right, we can't freeze them out. Could you, I think you did this, maybe this is before, it looks like it is based on the time. Could you provide the best place and way to report sightings or send photos?
Yep, just go to agr.wa.gov/hornets. There's a reporting form right on there, or send us an email.
And in Oregon, report to Oregon Department of Agriculture, you said?
Yep. Yep.
Okay. So just a couple more questions coming in here. The map of Washington State that you showed orange dots of reports in the Southwest region. Which cities, counties were these reports in? These would be unconfirmed reports, right?
Yep. So anyone from Washington can report a sighting of an Asian giant hornet. If they report on our online reporting form, they can mark it on a map where that report is coming from. So we do keep all those reports. We log every single report we get in. So if it's not confirmed, it'll be orange.
Okay. Is it known how Asian giant hornets were first brought to North America?
So it's unfortunately just speculation. You know, we didn't watch Asian giant hornets fly over here or come over here. It's speculated that since, you know, imports and exports are really large, that potentially they came over with some type of import. There's a couple other speculations. Asian giant hornet I mentioned is a delicacy, so potentially brought over for some type of food, and then, you know, it went bad. But most likely, since queens can potentially overwinter, you know, a hornet might not have been flying around in cargo or in freight. It might have been overwintering and then got loose.
Okay, I don't see any more questions, but send them through, people. We have a little more time. I'm gonna ask a question. You showed like the lookalikes, and there were some that were surprisingly like pretty much the same size. Are those, you showed stuff that's not from this region though, right? Some of those are not from Washington, Oregon?
Actually, all the insects that I showed, you kind of will see in the Pacific Northwest. Some of them, most of them are found throughout the United States. The one that can be up to 2 inches long are cicada killers. They're very commonly reported because they, you know, they have stripes. They can be pretty large. Usually the size is what will deter Asian giant hornets from any other insect, but cicada killers can potentially get pretty big.
But if you see one of these, you're gonna know like, wow, that's a really big bee type
The best
thing.
thing to do is just if you can snap a picture and report it, that's the best way to confirm or deny if it's Asian giant hornet. It's just report.
Great. All right, so Courtney threw in the chat box. If you have any additional questions for Cassie, you can email her. There's her email in the chat box. Everyone should be able to see that. I guess we have one more question that came in. Are they aggressively responsive to sounds and vibrations, such as mowers and trimmers?
So if an Asian giant hornet's just out flying around, they're typically non-aggressive. It's going to be if you come upon a nest. So if a mower or something, you go to mow upon a nest, the same thing. It would be the same thing as walking upon a nest. So if you typically, like with any other insect, if you leave them alone, they'll leave you alone, but they will defend their home.
Okay, here's a question that maybe is related to them defending. How easy is it to take a picture of an insect like that?
You know, keep yourself safe, but if an insect's like resting on a leaf, go ahead and snap a picture. If an insect's dead, go ahead and snap a picture. Just keep yourself safe.
All right. That's all the questions I see in here, so I'm going to kick it back to Troy. Thank you.
Great, thanks, Lucas, and thanks, Cassie. Really interesting stuff. I think, you know, maybe one silver lining we've all enjoyed is that invasive species have been in the news, you know, repeatedly because of the Asian giant hornets, and it's helped segue us into a lot of other conversations for other invasive species. So that's awesome. We are now going to break for lunch, and we will reconvene at 1 o'clock sharp. We are getting out of here right on time. So we'll see you all in an hour. Well, we're hitting 1 o'clock here. Hope everyone got a good lunch. I just put down 2 cans of sardines and about a half a brick of Philadelphia. So I'm pumped up and ready to go. Those crackers too, those flip side crackers, they're like a pretzel on one side and a Ritz on the other side. Perfect. We're going to go ahead and get on to the next part of our presentations, but first we wanted to announce the winners of the Kahoot quiz. So I'm going to pass it over to Courtney for that real quick.
Yeah, thanks, Troy. So it looks like our third place winner is Conrad Cower with Benton County Noxious Weed Control Board. And our second place winner is Justin Bush with the Washington Invasive Species Council. And our first place winner, drum roll, is Harris Kidd with Clark County Vegetation Management. So congrats everyone, and thanks to everyone else for playing along with us. I have your addresses, so I'll go ahead and mail those prizes out to you guys when I can. So
thank you, Courtney. Our next speaker is Dr. Rachel Meyer from University of California at Santa Cruz. And the title of her presentation is A Public Environmental DNA Database Helps Reveal Biotic Linkages to Invasive Species. Rachel is originally from the Pacific Northwest and trained in genomics, genetics, botany, and agroecology at the New York Botanical Garden for her PhD and at New York University for her postdoc. She then worked as an AAAS Science and Technology Policy Fellow at the National Science Foundation and was inspired to focus on an emerging tool for tracking life on Earth, and that is environmental DNA. She joined the University of California to direct the UC Conservation Genomics Consortium, which launched the California Environmental DNA Program, otherwise known as Cal-eDNA. Welcome, Dr. Rachel Meyer. Thanks for being here.
Thank you, Troy. Hi, everyone. I'm really excited to be able to present today, and I've been really inspired by all of the presentations and learned so much in the Kahoot quiz. So thanks again for having me here. All right, you can see my main screen, I hope.
Yeah, that looks good, Rachel. Thanks.
Cool. So I'm gonna just get going. So I'll be talking about environmental DNA today, and this is something that really excited me. About 5 years ago when I first learned about it, I thought maybe it could fill in a lot of gaps about biodiversity, kind of help standardize biodiversity monitoring to complement traditional observations so that we can get more biodiversity data and help model biodiversity change. So environmental DNA, you can just scoop up some soil or water or sediment. You can get environmental DNA out of air. You could get it off of the microphone or your keyboard. It's very like Gattaca-ish, right? You can take an underwater sense robot and collect it and like sort of filter water, and then take the DNA from the water and reconstruct, like sort of forensically reconstruct like everything that lived in that kelp forest that you just sent that little robot through. Or you can go into ancient sediments and caves and find find relic ancient DNA that can help you reconstruct what that habitat used to look like. So it's an exciting tool. There's a ton of technical work that we need to do to actually get it effectively into the hands of managers. And why I'm excited to present to you today is this is our first really big project where we've been working with preserve managers with environmental DNA to try to sort of see how it fits into the repertoire of what they're doing. Okay, so I use a technique called environmental DNA meta barcoding, and this is based on DNA barcoding, which is where we take different pieces of DNA, like the CO1 gene from the mitochondrial genome, or site B or 12S, these different genes that vary species by species, just like a unique barcode. We amplify them with primers that are in very conserved regions of the genome where there's never any variation, but these primers span these genes where they have a lot of variation from species to species. So they're a very good diagnostic tool. If you detect a certain sequence of DNA, you can say, oh, I probably have Channel Island fox in this sample, for example. So with next generation sequencing technology, you can sequence thousands of things at the same time, or tens of thousands, even millions of different DNA reads at the same time from a single mixed sample, and then parse out from your meta barcoding data all of the different species that were in that sample. And so this is sort of a macro view of what eDNA meta barcoding data look like. This is for the 18S locus that detects general eukaryotes, not necessarily to species level. This one's more like better resolved to family level, but very broad biodiversity. These are data from the Russian River. And what you can glean from this sort of presence-absence pattern across 172 sites that are on the x-axis, taxa are on the y-axis, is that sometimes you see almost like a line. And that means that that taxon, maybe that species, is almost ubiquitous in the environment. It's always present. And sometimes you see sort of empty white space. And that's probably a taxon that's pretty rare. So you're pretty lucky to detect it. Maybe these patterns of frequency that we find something can be very useful to us. So the Cal-eDNA program was meant to be really run by teams of citizen scientists or members of the public and university or other types of researchers. so that these partnerships, we try to build a feedback loop where we're getting help collecting samples. We're also trying to broaden the number of people, of brains, right? We just need lots of brain power to think about eDNA and what it can do for society and how we can improve these measures of biodiversity. So it kind of brings in more thinking about environmental DNA. So we started this program in 2017 with some funding from the University of California. People go to our website, they sign up for BioBlitzes, we send them out with gloves, and we tell them which preserve to go to where we have permits to collect, and they'll collect a small sample, send it back to us, we analyze it in the lab, or we just store it to build up collections that we can go to later as research resources. And we put the data online so that people can explore results and then email us anything that's interesting that they seem to find. So we do these not just single BioBlitzes, but we sometimes do coordinated sampling. That's what's really fun about working with the public, right? We need to go to 40 beaches in 2 days. That would be impossible to do as a single lab, but we can have this distributed sampling by working with broader teams. And we've had several undergraduate courses and modules to follow. Here's just a short cartoon of what the fieldwork experience is. So we give you a kit with different supplies, and you can scan your tube, which has a barcode on it that gives it a precise geolocation. That's what the tubes look like. We put the tubes back in a whirl pack so that they don't muck up everything. And you can collect abiotic data sometimes, or you just take a picture with your phone and upload a photograph so that we can kind of ground truth what you found. We also ask you to make iNaturalist observations. We use those to kind of complement the environmental DNA results. So sometimes it's nice to say, okay, we got oak DNA and you were standing next to an oak tree when you collected the sample. When we take scientists out, they are usually much worse at sampling things than members of the public. You can see our technician who does lots of genomics struggling to wade through this tributary here in the upper left. So it's also really nice to just like branch out. But when the samples come back to the lab, everyone's super giddy and we're excited to process them. It's a very detailed lab protocol, but well worth it because we get lots of biodiversity. So from this Russian River sampling event that I illustrated earlier with like that little kind of presence absence block, we had 172 sites and we detected over 16,000 different taxa. Now, this is just like hypotheses. They're taxonomic hypotheses. It's the best guess of what we think we found. So the closest match of something in the database. We haven't sequenced all of the tree of life yet, but we get lots and lots of fungi. We get lots of invertebrates and vertebrates, brown and yellow algae, green algae, and land plants, and protozoa and chromista. So really cool stuff and in different frequencies. So green here means they're very common to find, and yellow would be that they're very rare. Sometimes we get things that we cannot really explain that are like one-offs from certain sites. So in this case, one of my Russian River sites, this is in California, great wine tasting, had a freaking giraffe in it. And I was like, what is going on? It's not supposed to be here. But it turns out that just upstream, a couple kilometers northeast of our sampling site down here at Mark West Creek is Safari West. When you look that up, there is a giraffe image that comes up. And I love using this example because it kind of highlights the sensitivity of environmental DNA and that you have both a mixture of what's actually in that local community and things that could have blown in or, you know, traveled through a river in to your site. So it's kind of messy data, but it's big data. So we take all of this, we put everything online pre-publication. We don't wait for scientists to you know, slowly slog through the publication process because that delays sharing data with the public who contributed their time and effort to this by sometimes years. So we now have many thousands of sites that you can actually like order. You can check out like a library, like you can check out a soil sample and you can see the organisms that we've identified. So if you study tardigrades, you can see which samples we have from where in the world. Most of them are from California because that's where the program started, but we would love to develop partnerships in Washington State. Really would like that. If we can do Baja through Washington, like into even like British Columbia, that would be so rad. make so many cool discoveries. So you can actually like check out that sample and then sequence the tardigrades in it like more deeply and get cool tardigrade data. That's the idea. And so you can peruse our website, you can type in your favorite species, and you can see if we've detected it with environmental DNA. Tons of disclaimers, right? It shows that we have wolf. Do we have any wolves in California? Hell no, we killed them all. It's dog, but the same Latin name for the DNA sequence. So there's a lot to sort of filter through, but if we can learn to be literate in eDNA, I think we have a very powerful tool. So going back to this Russian River example, we have eucalyptus, and this is this dark purple in lots of our sites. So the sites are here, in this case, bars, and this is a bar plot of just like the relative abundance of DNA that we found from different types of organisms. So sometimes you get lucky and you get a lot of samples that have one species in it that you're maybe interested in because it's invasive and you want to understand more about its ecological role and maybe its partners. So here we can actually find in co-occurrence networks that we can make quite easily. I use a program called Speak Easy for this. Happy to nerd out about network analysis programs after this anytime. And I can see the fungal partners and bacterial partners and maybe even other plant and animal partners of eucalyptus that co-occur. So when I see eucalyptus, I often see these other species. And so that can help give me an idea of like how eucalyptus is interacting with its environment. For the Russian River, we found a lot of Ludwigia, evening primrose family that is invasive aquatic plant. It grows really fast, and then it tends to sort of get like this biofilm and other things accumulating on it that cause eutrophication. The plant dies, and a lot of habitat gets destroyed alongside it. So we've done co-occurrence networks for that. Basically, if you look at our website and you find any species where we have over 30 observations out of our total observations, we can generate a co-occurrence network for it. But there's sometimes, right, for some reason, Ailanthus altissima, there's gobs of it everywhere. But for some reason, because these are opportunistic samples, often maybe volunteers don't necessarily go towards the invasive species. They might go towards something that's maybe a little bit more iconic. I have no idea how they're making their decisions in the field collecting these samples, but we only have one site so far that has Tree of Heaven. So we cannot make a co-occurrence network for that yet, where we rely solely on the eDNA. So we need more samples in our database to be able to take advantage of this. But I just want to pause for a second and think, what else can we ask with co-occurrence information if we build up these databases? So you can put that in the chat or ask as a question. You know what, I think we can do this. This could be useful for me in my own research, my own management projects. Here's one example of a wildfire project where we had samples from before and after. Just the different colors represent different time points. After the Woolsey fire in Southern California that really affected the Santa Monica Mountains with some very high intensity burn sites. So we can actually like look at the species that are there before and after, and if they have the same partners in their network, or if they have different partners. So we can look across space, we can also look across time. So I'm gonna switch gears now and tell you, I'm hoping to excite you about co-occurrence networks and about environmental DNA and its potential. And now I'm gonna tell you an example where environmental DNA had a lot of limitations, but then we found a way to still do some network analyses that were pretty cool. Or we're hopeful about them. And this is about actually a really special place that's near my hometown of Palm Desert, California. These are desert oases that are managed by the Coachella Valley Nature Preserve from the Center of Natural Lands Management. So these oases you can visit. This is the Thousand Palms Oasis. There's a palm house. Sorry, it's the only picture I could find online of it. I've never taken a photo myself, and I should have. It's a house that's like built in the 1800s, completely made out of palm trees. Really rad. So if it was built like where the palm trees were stacked laterally like logs, it would completely flatten, right? But they were brilliantly building with the palm trees as vertical structural units, and so the palm house still holds. And this is actually where it's right on the San Andreas Fault, this oasis. So this is actually where people have been measuring the large earthquakes over time and how they know that like the big one is coming soon. There's lots of like ramps where you can walk through these amazing sulfuric oases. This is a neighboring oasis called Simone Pond. So 1,000 Palms Oasis and Simone Pond are about a 5-minute drive from each other. The 1,000 Palms Oasis actually is kind of the headwaters of Simone Pond. So there's the water underground continues into Simone Pond, and this oasis has a lot more light compared to the other one. And it's actually a place where they want to introduce the desert pupfish, which was never native there, but it's just the right habitat. So they tried to introduce it and it didn't thrive. And the reason for that is in the past is that in the 60s, the Boy Scouts of America gave a gift to the oasis managers of, the gift was dumping crayfish and tilapia into Simone Pond. And they thought this will just be great. It'll be a nice food source for people. People can do sport fishing and stuff like that. And they'll control each other because the tilapia will eat the crayfish and the crayfish will eat some of the food that tilapia needs, so they'll regulate each other's populations. And as you know, that is absolutely not what happened. The crayfish were in the more shallow areas around the perimeter of the oasis, and the tilapia in the deeper areas. They didn't eat each other, they just avoided each other, and they ate everything else, and really hurt this beautiful system. Here's an image of the crayfish that you can just see by staring into Simone Pond. And so the plan was to eradicate these. They were going to drain the oases, eradicate them with a neurotoxin, a crayfish-specific neurotoxin, and then put the water back in the oasis, continue to poison the oasis with this neurotoxin for 6 months until they had gone 100 days when they don't see any crayfish anymore. And then we just sort of keep monitoring and hope that the crayfish don't come back. But we would watch biodiversity kind of return to the area and ask, when are we ready to reintroduce it or to introduce the desert pupfish? So we went to kind of complement the trapping that they were going to do and this eradication effort. Right before they did the eradication, like just a couple of days before they started draining the oasis and adding the toxin, we collected lots of soil sediment and water samples. We collect them in these 1-liter IV bags for the water. It was pretty cool. We get to kayak in oases, and we have, this is a volunteer from actually like my high school that I got to like call up and be like, hey, can you come out and help us out? And we've continued to have volunteers, just a handful at a time, every time that we've done this sampling, even in the pandemic, which we're very lucky that we've been able to continue to do this work when a lot of fieldwork has been so strained. So we collected about 30 samples from 1,000 Palms Oasis. This is in June 2019, just a week before they started to eradicate them. And then 30 samples from Simone Pond, which had the crayfish and tilapia. This had there's other invasives here, but I'm not going to get into it. These are the major ones. And then we also made these experimental tanks because we had heard that crayfish DNA was sort of difficult to detect, and we wanted to be really sure that if we're going to understand like the eDNA signature of crayfish, that not only do we get them from the oases, but let's put crayfish in a tank and let them sit there for 2 weeks without changing the water. Then we'll take the eDNA from the water and we'll see what we get.
Hey, Rachel.
Yes.
5-minute warning.
Thank you. Perfect. I have myself on a timer as well.
All right.
So we have 3 crayfish tanks, 3 crayfish and tilapia tanks, 3 tilapia tanks, and 3 control tanks. And like we suspected, crayfish are awful little, and they barely shed any DNA for us to detect. Even though we knew over 26,000 crayfish were in Simone Pond, only 6% of our samples that should have had crayfish had any crayfish DNA in our metabarcoding results. They're very difficult to detect, as reported in other environmental DNA studies. And any species-specific assay kind of adds a lot of cost, and we found isn't even very accurate. So what we're trying to do is this different approach. We're going to say, okay, in places that we know the crayfish exist, let's find what environmental DNA-based taxa are enriched in those areas so that we can call that a putative crayfish holobiome. Let's mathematically figure this out. We saw people had published the crayfish holobiome from actually like swabbing crayfish gills and their guts and their exoskeletons to see what microbes are associated with them, assuming that they're on their bodies, they do filtering from the environment. So you have just the general biota of the environment, but there's some host filtering, and you get like a sort of an enriched community around the crayfish. So the crayfish have this also in the other direction too, where they are affecting their environment and shaping the holobiome of the entire oasis. So we were very hopeful about this. Here's a lot of bacteria, enterobacteria, alpha proteobacteria that have been associated with crayfish in some recent literature. So we had very different biodiversity in the pristine oasis, the invaded oasis, and the control tanks that we used in different statistical analyses. We actually have a third one. So we t-test, SIMPR analyses, and Solbol, which is like an R package that looks at beta diversity. And we would just, compare groups. Okay, these are all the samples that have crayfish and these don't. And we found lots of taxa that were very common in Simone Pond, never found in the pristine oasis, were also in the tanks, and some of them even had literature to support their presence. And so this is our draft holobiome. We have 90 taxa. Many of them are not reported in crayfish. The Hypocreales, this was really exciting for us to see. These are actually the taxa that infect crayfish to cause that like black spot, whatever rot infection thing. I'm a botanist trying to talk about an invertebrate, it's weird. And so we have these 90 taxa, and we're like, that seems like a lot of bioindicators. How do we whittle that down a little bit? Or how do we understand, like, how can we weigh them? Can we score these bioindicators for how potentially useful or interesting they might be? So we did that. We looked at the entire Cali DNA database that had 1,000 species in it, and we asked how many of these bioindicators, how many samples that other people have found from other parts of California have these bioindicators. So if that species we think is high in Simone Pond, but it's actually it's high in 50% of California, where most of those places don't have crayfish, then it's not a very useful bioindicator. We just got unlucky, and it somehow was statistically significant in our sample set. So we also looked if they were high in Simone Pond, where the crayfish were present, and then got lower, because the other way around, you could have a bioindicator, but it wouldn't be very useful to us. We looked at the geographic range, if they were pretty much restricted to the Coachella Valley where the oases were or not. And then we looked and did a lot of literature review to look and see if the crayfish were associated with those bioindicators, like from that study of the microbiome, the gut microbiome that I just showed you. And we find that, and we also looked to see if they were significantly associated in multiple types of statistical tests. So we've scored these bioindicators, and then now we're doing this post-eradication sampling, and we're checking if those bioindicators are going up or if they're going down. And we're trying to sort of understand, okay, across this scoring system, what is the pattern that we see? So we have 3 more sampling periods after the June pre-eradication in June 2019, December 2019, June 2020, and December 2020. Yeah, sorry, I should have changed the order. So this is just a general heat map. It's very ugly. I'm still working on a way to figure out how to make this beautiful for all of you to see. But what we noticed was seasonality makes things complicated. Things that were never there in June or there in low numbers maybe are very high in December, because man, December and June are pretty damn different seasons in the desert, right? And some taxa that were bioindicators, they're high here in 2019 before the eradication, they disappear in December and then they come back. So we're actually, there's still some bioindicators that have gone down and never returned, but we really think that just annual tracking at the time point the most appropriate time point when you did develop those bioindicators, that's what we should stick with. And our next step is to add all of these data to this global biotic interactions database so that other people can take advantage of it, just like we took advantage of other studies. So what it all means is my final slide, is that eDNA provides another tool in the biomonitoring tool belt. It's not an end-all answer to anything. It's still very hairy to dig through. And the only real way to see if the eDNA holobiome bioindicators work for monitoring is to put them in a natural experiment. But again, we don't want to put crayfish back and see if they work. That would just be a pain in the neck to eradicate them all over again. And there's a lot of potential false leads because of how common species are. If you can't compare to other studies, it would be a lot more difficult to find trustable bioindicators. So remember, invasive species, really nice to be able to eat them. Unfortunately, these crayfish, you couldn't just say, let's just boil the oasis and have a big party because they taste like sulfur and they're horrible. And I'd like to say thank you for your time and for paying attention to this presentation. I'm very grateful to my 2 lab techs that processed the data and to Kim Belair, who's been leading the study. She's a postdoc here at UC Santa Cruz and was in some of the earlier photos. The end.
Great, thanks. We got a little bit of time here for questions. And then as we've done the other ones, we'll pass on your email so people can email you with questions. I'm sure you'll get a lot. The first question is, are you interested in and can you help with the processing and analysis of samples from Western Oregon?
Yes, yes, but there's a pipette tip shortage right now. So if the projects are not urgent, then yes.
Well, that's great. And then I think we got a lot of people throwing out what, in response to your co-current question, what could be looked at. Someone said indicator species. Someone said you could compare species occurrence over time with pollutants and how the populations are affected.
I like that one. Yeah, cool.
Are species changing with climate change and how? And another question, is limitation of eDNA availability of a reference DNA sample in the database? Did I read that right? It's really small. Is limitation of eDNA availability of a reference DNA sample in the database? Does that make sense to you?
That is definitely one of the major limitations. We only have we have fewer than half of the species, even like big things that you can see and easily visually track sequenced yet. But 90%. Pardon?
Sorry. Their secondary question was, would eDNA detect only known species or everything in the sample? I think you answered that.
It could detect everything in the sample, but you might just have the DNA of it and not know what it is. But you could just say, I have this unique thing. So you can still count things. You just can't name things all the time.
All right. Someone says your earrings are amazing.
Thank you. They're cicadas. I only wear them once every 7 years.
A lot of general excitement. I think you'll be able to see these all too. And the last one I'll ask is, when are you collecting samples? Is there a specific season/time period?
We try to do a spring and a fall, like big BioBlitz, where we hit up as many reserves as we can. So this April, it would be really great to be able to coordinate sampling if anyone's going out.
Cool.
Thank you.
Well, thank you. Yeah, so if you have any more questions, you can email Rachel. Courtney just put her email in the chat box, and I'll hand it back over to Troy. Thanks, Rachel.
Thanks. Thanks, everyone.
That was great. Yeah, thank you, Rachel. I bet your email is about to blow up, so be ready. Our next speaker is Jennifer Gervais, and she is going to give a talk called Climate-Induced Expansions of Invasive Species in the Pacific Northwest. Jen is a wildlife ecologist for the small nonprofit group Oregon Wildlife Institute, and she has worked on research and conservation management projects on a wide variety of subjects for 30 years. She has worked on projects involving burrowing owls, gray-tailed wolves, western pond turtles, Oregon spotted frogs, and many other species. One of her more recent projects involved assessing what we know about invasive species and climate change in collaboration with the research team from the USGS aquatic ecologists. So welcome, Jen. Thank you for being here.
Hi, let's see, I'm trying to show my screen. Did that work?
It did. Looks good.
Okay, great. And I am realizing that I'm going to be looking a little bit to my left because I managed to mess this up a little bit, but hopefully that won't be too much of a problem. So I'm discussing some other research today. This was a literature research, and it's really hard to follow the previous presentation because Rachel's work is, in my opinion, a lot more interesting than what I'm going to tell you about. But my colleagues and I wanted to know what exactly we already do know about climate change and invasive species. And this presentation is a summary of the research that we conducted and published last year. So you can find the citation of the paper itself and biological invasions at the bottom of the screen here. And so everything I say pretty much is in this paper for your reference. If you this is the Cliff Notes version. You no longer have to go and read this thing. Okay, let me see if this is going to let me do this. It will if I get my cursor in the right place. There we go. So invasive species and climate change are both known to be enormous issues. They both drive a tremendous amount of change in biological systems. But so far, it's not been clear how much we've examined how much climate change impacts will facilitate or change invasive species impacts. And so this question came up, and this grant was to try to explore what we've actually know already about how these two forces might be interacting. So what we were looking at were patterns of range shifts, because we wanted to approach this issue with more of a management issue, emphasis. And so our questions were, what range shifts have been documented within the scope of climate change? And are there projected patterns of range shifts that have been suggested under climate change that might help us evaluate what might happen next? And in particular, we were interested in discovering whether or not there might be patterns out there that could either guide management actions on the ground now, or at least identify future research needs that could later inform management policy as we struggle with the issue of climate change and particularly invasive species. So we limited our region of interest to the Pacific Northwest. And for our purposes, we defined that as Oregon, Washington, Idaho, Western Montana, which basically included the Rockies to the west, and the southernmost British Columbia. And this area obviously includes an enormous range of ecosystems, and so it's kind of a microcosm for the rest of the continent. It's also an area of rapid human population growth. Some of the most rapidly growing cities in this area are actually east of the Cascades. And increasingly in this region, we are seeing rapid changes in climate and disturbance regimes. So the first step was, what are we going to ask? And we determined that we were going to look at geographic patterns over mechanism because we wanted to look at more broad scale things. And because of this, we didn't look at mesocosm studies or other manipulated experiments, although those are critical to understanding mechanisms. And we also didn't address ecosystem vulnerability to invasion or what characteristics might identify a potentially successful invader. Again, there are vast literatures already on those subjects, and we didn't feel that revisiting that fit within the scope of what we were able to do. We also stuck to natural systems and did not look at literature or species that were primarily an issue in highly managed ecosystems, such as agriculture. Again, we had to draw some boundaries somewhere. Our second step was to identify what species we were going to ask whether or not climate change may have facilitated their spread. We decided to stick with terrestrial and freshwater species because we were all aquatic or terrestrial biologists. And we chose to exclude disease organisms, microorganisms, parasites, or their vectors. These things are primarily looked at from a human health perspective, and not to say that disease and parasites don't have an enormous impact on ecology. We know that they do, but there's a little bit less information about those. So we stayed away from those guys. We also didn't look at native species whose ranges may have expanded without help. So for example, the barred owl was out. We did not look at game species, and we excluded species from consideration if they were managed for any other purpose or value. And an example with this would be the wild horse, because there are a great deal of other values in those besides just their issues as an invasive species that are going to influence their management and therefore would show up in their distribution, abundance, spread, or impact. And then finally, we only looked at species that were either established in the Pacific Northwest already or were considered a critical invasion threat. In other words, the conditions exist right now for those things to potentially establish and to flourish. So species identification, who's on our list? For the aquatic species, the guys got off easy. The USGS non-indigenous aquatic species database is sort of a one-stop shop for all of your invasive species listing needs, because it includes organisms from every taxa that are an issue in freshwater ecosystems in North America. The terrestrial species, however, were much more problematic. We used state's noxious weed lists. That was the easiest part. I scoured state wildlife agency websites to get a feel for what vertebrates were an issue, but again, excluding game species and organisms such as wild horses or burros. And then I took any other list I could find to get a representative sample of insects of concern. And this last was really surprising to me, that I was not able to locate some kind of comprehensive list of insects that are of concern to terrestrial natural ecosystems, that it was really hard to identify them, and that really surprised me. We also looked at the Western Governors List for the 50 worst invasives and the world's worst 100 invasives list. And from those lists, we took species that were either already in the Northwest or could potentially successfully invade right now. So here's the species list we came up with. Half the species we looked at, a little bit more than half were plants, so then that partly reflects the quality of the lists that were available to us. But we still got in 82 species of ray-finned fishes, 13 crustaceans, 12 bivalves and snails, 2 bryozoans, 2 hydrozoans, 63 insects, 6 birds, 6 reptiles, 5 amphibians, and 3 mammals. Only 3, because most of them really are managed for something else, for a total of 2 kingdoms, 11 classes, and 398 species. And so we had a nice taxonomic representation, although I will note that the microorganisms are not here. We chose 27 species from the 100 worst invasives global list, and we took 48 from the 50 of the Western Governors Association list. Thankfully, coqui are not an immediate invasion list risk here, and let's hope they never do become one. So for our methods, we used Web of Science, and we searched the literature from November 2017 to March 18th. And I am aware that some very relevant papers were published almost immediately afterwards, like they probably crossed in review kind of a thing. But that is the window from which we drew. We searched Web of Science using Latin name or common name and climate change and distribution or abundance or spread or impact. Again, looking for patterns. Plants in particularly are prone to getting reorganized taxonomically, and several times there were individual species had their genus changed. And when that was true, we did searches on all combinations of the Latin names and common names to make sure that we picked up everything in the literature that we could. We retained studies with climate change or synonyms in the title, abstract, or keywords. So climate change, global warming were all things that we picked up on. Global change was one that we saw frequently. And then once we had a hit, we would take that paper and extract information from the methods and results. So for each study, we recorded the primary drivers of the invasives dynamics, such as precipitation, temperature, or stream flow, or whatever else it identified. We recorded what the range dynamics that the paper discussed were, for example, demography or impact with respect to those drivers. We determined whether it was a documented change or whether it was a predicted change, a prospective study. And then we noted whether it had occurred within the species native range and whether or not the study had actually occurred within the Pacific Northwest. And what we were originally hoping to do was to do some taxonomic and ecosystem comparisons to see if we could find out with some larger patterns that might actually help management evaluate and anticipate what might be happening in the near future. Well, we very quickly found out that despite the fact we were looking at so many different species and we had such broad searches, that there were some serious issues with the data. For one thing, the papers varied tremendously in their methodology and also on their output. For example, some might only give qualitative maps, others might give very quantitative changes in range. And so that it became very hard to compare from one study to another. Most of the studies we found were projections of climate models and habitat suitability models. And so it really depended on what climate projection they chose to use and what habitat suitability model. And it was very clear that you can have different answers depending on which model you use. And many of these studies, out of necessity, were extrapolating from large-scale climate projections because that was what was available at the time. So we were really forced very quickly to realize that we could only look at qualitative comparisons. But we pressed on, hoping again we could find some kinds of patterns here that could guide us going forward. And the first shock was, despite the thousands of searches that we did, very few studies have actually documented actual changes in the distribution, abundance, spread, or impact of invasive species through the lens of climate change. We found a whopping 10 studies for 157 aquatic invasive organisms. And of these 10, 3 actually were addressing changes seen in the Pacific Northwest. Only 5 studies were found for 241 terrestrial invasive species documenting change. And of these 5, 2 addressed change in the Pacific Northwest. Of the Western Governors List, again, we looked at 48 species in that review, and only 4 of them have actually been studied before. So we don't even know anything about the other 44. And of the 100 worst invasives list, we included 27 in the review, and of those, 5 have been studied. So tremendous lack of knowledge about a lot of the organisms that we've already identified as being of critical concern. So what did we find? Of the 3 studies conducted within the Northwest, all of which were by my colleagues, incidentally, all 3 examined hybridization of native West Slope cutthroat trout and invasive rainbow trout. And what they found was that the hybridization between these 2 species increased with declining spring precipitation and spring flow and increasing summer water temperatures. And so even if the invasive species wasn't actually moving, the genetic material was, which was an interesting pattern, an interesting subtlety perhaps that we hadn't thought about before. And it would be really interesting in light of Rachel's work, what the eDNA would look like with a hybrid. So for aquatic studies outside the Pacific Northwest, there were 3 studies out there that have existed examining the expansion of the centrarchid fishes. And what they found in Ontario was that the basses and the bluegill sunfish did expand northward. They had actually documented that change. And at a smaller scale study, regional expansion was examined. And some of those same species were found to expand northward generally, but it was really spotty. And the authors concluded that biotic communities were still influencing establishment, even in the larger picture of northward expansion. The third study looked at green sunfish in California, I believe in the Central Valley. And what it found was that the green sunfish were able to establish during a super seasonal drought, in other words, a time of extreme stress to that ecosystem. And then they were able to hang on and continue existing there even after conditions returned to what had previously been considered normal. Outside of the Pacific Northwest, there was an incident where the common reed has been found to be spreading because of increased hurricane frequency, and that had to do with the salinity of the water declining in estuaries. This was our one borderline estuarine species. And so these changing storm frequencies was related to changes in water quality, which then influenced the species establishment. Carp, the common carp, was expanding in the Murray River drainage of Australia with stream flow reductions that were brought on by extreme drought. And interestingly, the carp expanded despite the predictions that they wouldn't be able to do that, but they were able again to maintain their range expansions once conditions returned to more normal conditions. Finally, there were 2 studies dealing with zebra mussels. One documented earlier spring spawning with changes in climate, and another one suggested that density was increasing with climate change as well. But further examination of that data made the authors conclude that those changes might have been mediated through trophic changes instead. And so how the role of climate change may have been playing in there was a little bit more nuanced. So for the terrestrial species, again, very few papers. We found 2 within the Pacific Northwest. The first one by Snow and colleagues documented that feral pigs have been expanding in the Pacific Northwest, and that it seems to be associated with milder winters with less snow cover. Gypsy moths have also been moving west into Washington and British Columbia. And this study actually didn't look at simply temperature as a single light entity, but modeled it as a sort of a habitat suitability matrix. It looked at extremes of temperature and durations of different temperature regimes, and found that temperature suitability is increasing for the species in this area, and that the gypsy moth, not surprisingly, is moving with that suitability. For terrestrial species beyond the Pacific Northwest, we had 3 examples. The house sparrow has been established within North America for many decades, and it has pretty much completely covered the country. However, finer scale distribution changes appear to be going on with interannual long-term temperatures that are modifying the species temperature niche. Gypsy moths are changing their range in Virginia. And this ended up being a really interesting paper because the super optimal temperature duration was really the key here. So super optimal temperature is above what is normally considered best for development, but a tiny bit over that would accelerate the development to the point where the gypsy moths could really expand very rapidly. Raise it just a tiny bit before that, above that, and it was lethal. And so they saw these shifts rather than expansions. As the animals were killed out of areas that got hotter, but then there were areas where temperatures became more suitable and they expanded into that. A final study looking at potato tuber moths actually in the Korean Peninsula found that it appears that the potato tuber moth is increasing with increasing air temperature. So not a lot of detail on that one. So what do we summarize from all of that? We know in the aquatic systems that climate change has indeed mediated invasive species expansion, and increasing air temperatures and decreasing precipitation were the 2 indicators of those changes. For the terrestrial species, climate change was associated with both the increasing and limiting range expansion by non-native species. And so it was a little bit more nuanced in that case, again, because of the direct temperature relationships with insect development. So from the prospective studies that we looked at, we found far more of these than we did actually documented results. 93 total studies looked at 117 species-specific predictions. And once you've gone through the work of setting up your climate map, it's a little less work to take your habitat suitability model and put it in there. So many of these papers looked at more than one species. So out of these 117 predictions, 43 were for aquatic organisms and 74 terrestrial. 30 studies overlap with the Pacific Northwest, but only 6 focused within the Pacific Northwest, and those 6 studies looked at 11 species, so still not a whole lot of data. There were no studies at all on 8 of the 27 species from the 100's worth list, or 27 of the 48 from Western Governor's list. And again, the general methodology of these was to use climate projections and ecological niche models. Quickly, we had 43 predictions for aquatic species, 26 in the Pacific Northwest. Fish were the majority. Surprising to me, even outnumbered plants. Crustaceans 8, 4 for mollusks, only 1 for the mammal, 1 for a reptile, and 1 for an amphibian. So there's a very strong taxonomic bias here in what we know. So I'm going to cover these. I see that I've got 10 minutes here, but especially because Rachel didn't have a chance to field many questions, I want to make sure I finish on time. So I'm going to go a little bit quickly through this. Only 2 species were the subject of more than 1 projection. And this really surprised us too, because we were hoping to be able to look at several studies and compare their output to give us some idea of pattern. You know, you can kind of think of each one as a data point, and that really wasn't possible. We don't know enough even with this type of modeling. Some species were projected to increase their range, some were to decrease, and 1 no change. So again, it's more nuanced than just climate change is going to make everything worse. Unfortunately, both mollusks, all 5 plants, and the nutria increase. And of the 4 species of fish, however, there was a little bit more of a mixed response. Rainbow, brown, and brook trout were all projected to decline. However, we know that hybridization between native and invasive brook trout is increasing in Montana. And so this suggests that we need to be pretty careful about just taking these prospective studies at face value. We have to realize they're just that, they're projections, and they're often based on fairly coarse data. And another caution is that we know brown trout are known to be increasing in Montana. So we know that some of these modeling studies are likely to be off. One of the other really interesting studies we found was a smallmouth bass study in John Day, Oregon. Lawrence and colleagues in an ecological applications paper in 2014 were projecting increases of bass. This is consistent with range expanses of the species in the Great Lakes region. We already discussed that. But what they did find that was interesting was that climate mitigation through riparian restoration might have a big impact on keeping these fish from interacting with the native salmonids in this river system. And so they concluded that extensive restoration could potentially limit upstream expansion of the bass, although they noted that the effect between different parts of the drainage varied quite widely. And whether they've done follow-up work, I'm not sure, but it would be interesting to explore that. For the prospective studies for the terrestrial world, we found 74 predictions for 47 terrestrial species, 24 plants, 21 insects, and 2 of the mammals. So a little bit better balance in the taxonomy. So the predictions for terrestrial non-native invasive species, we had 15 predictions of increase in 26 projections that overlapped the Pacific Northwest. 7 plants expand, 3 were static or declining. Of insects, 6 expanded, 4 were static or declining. And mammals, both species, alas, feral pig and a feral cat, are projected to increase. And the photograph here is of the cat that's probably busy dismantling my nest in the other room, who came in out of the cold. They're very adaptable. So species, spatially explicit terrestrial predictions include that cheatgrass is projected to be stable in the Columbia River Basin, but to increase in Idaho and western Montana. The yellow star thistle, rather similarly, is also predicted to expand in Idaho and western Montana. And all of these appear to be driven by declining precipitation and increasing summer temperatures. Interestingly, spotted knapweed was predicted more to shift from western Montana into Idaho, so it looked like more just a range shift. And there was very little change in the models projecting leafy spurge in the Pacific Northwest. So what have we learned? There's been so little research overall, it was really shocking. And we kept adding species and adding search terms, trying to see if we were missing anything. And we finally came to the conclusion that we're not. There's just not much known. Few species have been the subject of even one study, which makes any kind of meta-analysis absolutely impossible. Fewer still have been examined in more than one study. Climate change is likely to facilitate the expansion of many invasive species at broad geographic scales. There's going to be a general northward expansion, for example. But local conditions look like they're still going to be pretty critical in terms of establishment and persistence at local scales. And this is probably where management is going to be so critical. Prospective studies should be used as a basis for further inquiry rather than management actions. The cautionary statement there is about the fish where they were all projected to decline, and actually that's not what's happening on the ground, at least not yet. More fine-grained projections, both of climate maps and of local conditions, are likely going to greatly improve the relevance of these projections, and particularly if they are married with other sources of data, such as the work on mechanisms that has occurred at smaller scales. So with that, I have 6 minutes, awesome, for questions. And I have my email down there too. Feel free to email me if you have any questions, or if you'd like a copy of the paper, I can get that to you.
All right, thanks, Jennifer. Go through, we got several questions here. The first one, and I think Courtney's gonna share this. Someone commented there's a U.S. invasive species list. The USGS made a comprehensive list, all taxa, with references in 2018. It's a great resource. So Courtney's going to share that, it looks like.
Oh, great. That came out probably right after we needed that. Yeah, because we started in 2017. I want that. Thank you.
Yeah, looks like there's a lot of questions, or there's been other stuff in the chat too about different lists and stuff. So I think a lot of people have opinions on that, or thoughts or suggestions for you. A question here says, why did your group decide to limit your literature review to the date range selected?
Actually, that wasn't the date range we limited our literature review. That was the time period during which we searched. So if the paper was not published before 2017, or was not published, prior to 2017, we weren't going to pick it up in our search. And I know that there were a couple of papers that were in press at that time that we didn't pick up because those authors reached out to me. So we were limited somewhat on the funding constraints. It wasn't a large grant, and so we had to get it done in a fairly short period of time. And again, that is the window during which literature had to be available in order for us to find it. I hope that's more clear. We certainly went as far back as anything occurred in Google Scholar. It was just a matter of was it in Google Scholar at the time we did the search.
I see, yeah. That is clear, yes. Someone just says, very cool literature review study. So surprising that few studies have looked at biogeography changes related to climate change. I think that's one of your summaries, and we just don't have that much to look at. So one thing you found. Someone shared a paper, and Courtney can probably get this to you, but then maybe they say you already have it on nutria and climate change in the Pacific
Send it along.
Northwest.
If you have a citation,
Okay.
please send it along. I'd like to see that.
Yeah, someone
again, I'm aware that a number of studies were in press, and I'm also aware I missed one. I know that I missed one study on salt cedar because the author reached out and sort of went, how come I'm not in there? And unfortunately, I missed that one. But, you know, we didn't miss very much. I think the pattern of how little we know would not have been changed, even with one or two more studies that we might have overlooked in our search.
Okay. Yeah, so that full citation is here, so she can share that. Someone said, in your opinion, why do we have a governor's list of invasive species if the majority of those species have not been studied?
You know, that's a good question. I don't know really how to answer that. My guess is that these lists are primarily drawn up by people interested in invasive species who have heard about species causing huge problems elsewhere and then think, geez, that could be a problem here. We should list it, have it on our radar screen so that we're aware this one might be a problem. So I kind of look at these lists as red flag lists. Watch out for these things because they could be hugely problematic if they get here. We may not know a whole lot. You know, it's a good question. I don't know how those lists were put together, and I'm just guessing that it was people going, you know what, we don't know much about these species, but we think that this could be a problem.
Yeah, I think you're right. Please tell Jennifer this is a very interesting presentation, extremely useful. Someone else says, were the decreased precipitation findings / predictions based on conditions east of the Cascades?
Depended on the study, certainly were. The work on the invasive plants, the prospective studies, the Columbia Basin, for example, those were clearly east of the Cascades, and they were looking at projections of climate in that region. Bradley and colleagues published a number of species on that. If you give me an email, I can point out those references to you. But yes, some of those definitely were east of the Cascades.
Okay, yeah, and someone else had maybe a similar question based upon your answer. Someone said, what specific studies might you recommend? But no context beyond that.
Yeah, again, email me and I'm happy to share the paper with you, and you can get all of the citations of the literature we cited. And actually, I should say also that when you get this paper, get the supplement as well, and that will tell you the entire list of 398 species, whether or not we found any literature on it, either retrospective or prospective, and where we, you know, whether or not it was on an invasive plant species list, a governor's list, or the 100 worst invasives. So it's sort of a summary table. Biological Invasions did not want to publish a 17-page table, so it's the supplement. But I can send that to anybody who wants it.
Okay, and maybe someone else put a clarifying comment, possibly saying maybe they mean future studies. So are there any species that you recommend studying, I guess that person's implying?
You know, I really can't answer that question. What we really went at was just what do we know, and we don't know much at all. And depending on where you are and what your concern is, you're probably going to think different species are of priority interest. Fair enough, because again, the little bit of data we have suggests that local conditions matter. And they're also sometimes the hardest to figure out. I think from my own sense on this is that this should, I hope, be a starting point where people who have been looking at more mechanistic work at a smaller scale start talking to the more the biogeographic inch scale people so that we can start marrying the pattern and process and start generating some hypotheses that perhaps we can actually test first at small scales and then look and see what's happening. That hopefully will give managers knowledge of mechanisms that may also give you tools to stop some stuff.
Okay, well, great. Thank you so much. I'm going to pass back to Troy here.
All right, well, thank you very much, folks.
Yeah, thank you, Jen. That was really good. You know, I think so many of us have been thinking about climate change and not only identifying ways where we can think about some forward-thinking management that'll create more resilience as some of those patterns exist, but also what we can do to mitigate some of that. And it sounds like there's still some hope at the site scale where we're currently at. So that's great. We are going to take a break now, a 10-minute break. It is 2:01. Let's get back to 10 to 11, and we will carry on. All right, looks like Elaine has joined us. I'd like to introduce Elaine Harvey of the Confederated Tribes and Bands of the Yakima Nation. Elaine's going to give a presentation called First Foods and Their Sensitive Habitats. She's worked as a fish biologist for the Yakima Nation Fisheries Program in the Rock Creek Subbasin for the past 14 years. She also comes from the Rock Creek Kamilkpa Band. I hope I said that correctly. The Rock Creek Band is one of 14 tribes and bands that comprise the Yakima Nation. Elaine has a Bachelor of Science in Aquatic and Fisheries Science and a Master's of Science in Resource Management, and is currently a third-year doctorate student at the University of Idaho, go Vandals, in the Natural Resource Department, focusing her studies on climate change and impacts to Yakima traditional foods and medicine. She lived in this region her entire life and learned many of the traditional Yakima medicines and food sources from her late grandmother and other family members. All traditional Yakima foods are considered as medicinal foods and bring good health to the people. Her traditional knowledge, combined with her Western knowledge, brings about a concern for traditional plants and their resilience with climate change. As her elders and ancestors have had reverence for the land and the resources, Elaine carries those teachings and concerns for the environment for all future generations. Thank you, Elaine, and welcome.
Thank you all for having me today. And I was invited to talk about first foods and the sensitive habitats. And I come from the Willow-Whitespun Band and the Kamilkpa Band, and both of these bands come from the Columbia River. And they're located at near Maryhill, Washington, and near the John Day Dam is where the Rock Creek Band is from. And both of these bands, they are a part of the Yakima Nation. I wanted to talk about this statement in the Yakima Nation Treaty with the US government. The elders, our ancestors at the time, knew the importance of salmon and all our foods that we utilize throughout the year for ceremonies, subsistence. And it's a really important statement in regards to the first foods that we still utilize today, and where it reserves our exclusive right of taking fish and all the streams running through and bordering the reservation and secure to said Indians and all other usual and accustomed stations in common with the United States, and of erecting suitable houses for curing the same, and also includes the privilege of hunting, gathering roots and berries, and pasturing their stock on unclaimed lands in common with the citizens. And it's a really important statement that is in the treaty, and it assists us tribal members to continue our traditional lifestyle of fishing, hunting, and gathering. So here's a map of Washington State, and in the tan brownish color, you can see the Yakima Nation ceded lands, and these are lands that were ceded at the time of the Treaty of 1855. And other lands were also included in Oregon. Our bands traveled annually to Central Oregon to gather foods, so that's not included in this map, but in the green is the actual Yakima Reservation. And so throughout these lands that we ceded, we retained the right to fish, hunt, and gather. And so it's quite extensive, and I was personally think it's like almost a third of Washington State that we continue our traditional way of life. In this slide is a picture of the plateau tribes as referred to, the Yakima, the Umatilla, Warm Springs, and Nez Perce. You know, we're all neighboring tribes within the Columbia Basin. And as plateau tribes, we are similar in many ways. We have similar, our religion is similar. We all believe and belong to the Washat religion, also known as the Longhouse religion. And we gather many of the same foods. We all fish for salmon and the different aquatic species, and we all go gather the different roots and berries throughout this area that you see here. And these are seeded lands that in the different colors, the purple, blue, the darker brown and the green are seeded lands that the tribe ceded during the 1855 treaty that was in Walla Walla. And this is a picture here of our longhouse that we had at Rock Creek. Our band our band village is located 20 miles east of Goldendale, and there's a picture of our chief here with the war bonnet. He was Chief William Yellup, and he was one of our main chiefs who went out of his way to protect after the treaty, you know, the treaties were signed, but he went out of his way to protect our treaty rights for fishing, for gathering huckleberries in the national forest, and our hunting rights in Washington and Oregon. We still come to this place, the longhouse. In the lower right, you can see our longhouse. This is the modern longhouse that we utilize, but we still come here, we gather, and we honor our first foods. And so we have a long history in this region. Klickitat County is where we gathered a lot of our different foods, and even up on the reservation on Yakima County and then in Oregon. This slide is the seasonal round that we usually do every year, and it's been this way since time immemorial. Every spring we have the first fish that comes, and it is the bridge-lip sucker and the steelhead, and then we have the spring chinook and the other fish species that come up the Columbia, and we honor our traditional foods every year. It's ongoing, and all the different longhouses, as mentioned in the different tribes, we all gather in the spring for the first foods. So in the spring we have the bridge-lip sucker and the celery, Indian celery that we gather, and we have a feast to honor these foods, and we're not supposed to go and gather these foods until we have an official ceremony to honor, because there's a story that goes at the beginning of time for the Yakimas and the different tribes, the Columbia River tribes, that these foods, you know, stood up and they made that agreement with the Creator to give its life for the people that were coming, and the people were us. And so the salmon was the first, and all the different fish species that we use in the lamprey, and the different mammal and bird species, as well as the different plants, the roots that we use for food and medicine, and even the different kind of berries. There's all different kind of berries that we still go out and gather. And as you can see, this is also represents our Indian calendar. We knew when the food's coming. We knew when the salmon were going to be here. We knew when the different suckers were coming up the creek, and we knew when the different roots and the different berries were going to become ripe. That this represents our Indian calendar that's been here since for many years, and in our tribe, in our people, we know this as we're going to do this every year. We're going to go to a certain place and gather this certain kind of food every year. And I really think it's a really good picture that kind of explains where we, you know, kind of what we're gathering throughout the different seasons. And we gather foods in all different areas. We gather foods in the high meadows. We gather foods in the shrub steppe. We gather foods in the riparian area, and also in the alpines, where the berries are, where the different huckleberries and blueberries are, and that are up there. So there's all different kind of habitat types that we gather food in, and for us, it's really important to monitor these and to ensure that our foods will always be there for us. We have to monitor, and another important aspect is taking our children and our elders to these places, that we always continue to go to these places to gather foods. And because that is a teaching for us, is that you have to continue every year. We don't take time off, and it's just ongoing and continuous. And I hope and pray, my daughter and my children and relatives, we're going to continue this cycle of traveling to these different places. And it's really beautiful, all these different lands that we're able to access. In the upper left corner, you can see where the camas meadows are, and then the shrub steppe, the different areas where we gather our foods. And one of our teachings from the Longhouse religion is that everything is connected and everything has a purpose. When one species is left out, then it's going to affect something else. And we always honor, and I put these pictures in order because our way is everything's in order. The first is the water, and then our next is the salmon and the different fish species and the lamprey, and then the four-legged and the birds, and then we have the plant species and the berries. And as we go throughout our life, this is our seasonal round that we go through. And when we honor our foods, this is the order that we follow. And it's this is the way that it's been explained to us, and this is what we explain and carry on with our children. Here's another photo. I've done presentations before in person prior to the pandemic, about over a year ago. And I was able to go out and bring our traditional foods out to the presentation. And when we have our ceremonies and even any kind of gathering, we always put our salmon first, water, and then salmon, and then our deer meat and our roots and our berries. And that's just how we conduct and honor our foods, our traditional foods. And to the right is a picture from the Columbia River Intertribal Fisheries Commission, also just depicting the seasonal round that we believe in, that we follow. And it's the natural order of this land where we live. Besides for food purposes, a lot of the different plants we use for health and wellness. And it's kind of universal with other tribes. Other tribes, they utilize the same plants because they're some of these same, like the sage and the sumac, they grow in Arizona, in different parts of Nevada, in the stinging nettle and the milkweed. It's kind of, these are universal plants that grow in different Indian country. And also the choke cherries is kind of a universal berry that a lot of different tribes use, and they're all important to us for medicinal uses, for food purposes. And it's just, there's many different kind of plants that we gather and use all over. And as well as, like I mentioned, we gather our traditional foods and all over in alpine and shrub steppe habitat and different kind of habitats. That's riparian. That's how these medicines are also. And plant fibers, we use a lot of different plants and roots we continue to, this is part of our art, and it's part of, we use these as tools, like we have the baskets that are made from different plants, and then we have the huckleberry baskets in the middle. Our elders are holding. Those are cookatap baskets, and those are made of cedar root. And then on the right, we have the tule weeds that we use. We use them for a lot of different purposes. We use them for, at one time they were used for homes, for summer homes, and also they were used for in funerals, and also for when we have our ceremonies, we lay that on the floor, and our foods go on top of it. It's like our table. So there's a lot of different uses for plants and roots. And the tribes in this region, you know, they've always been resource and harvest managers of the foods, of fishing for salmon, for hunting, and for gathering the different foods. We knew that we couldn't over-harvest. We knew that we had to be caretakers of the land. And one method that was and has been used for many years and was fire, and it was used to replenish the huckleberry fields. Because today you go to the huckleberry fields and they're really overgrown by fir trees and the different kind of trees that grow, and they're overgrown and taking away the huckleberry habitat. So in the past, after the berry season was over, certain time of the year, certain people would stay there and start a fire and burn the huckleberry field, and that was just one method that for fire that was used. In the bottom right picture, you can see a picture of the huckleberry fields where it was burned, and then in the left bottom picture, you can see how open the huckleberry fields were at one time, and now you go to the huckleberry fields and all you see is a lot of fir trees, and they're taking over the habitat. And also fire was burned for meadows where the roots grew, and also for grazing for horses. And one thing when we go root gathering is we bring our roots back. We bring the peelings and the seeds back to where we gathered it. It's one teaching, and that's to bring the seeds back to where we collected the roots as a harvest management tool also. I'm a fisheries biologist, and I've done some fisheries restoration, and I've talked to some, and I've been questioned by some other fisheries ecologists or habitat ecologists and about restoration of streams and meadows. And I always reiterate that it's important, you know, to always utilize natural native plants of the area, because, you know, that's really what is supposed to be there for the wildlife, and also for our cultural uses that when we go gather we gather a lot of different medicines and food types and fibers from the creeks. In this photo, it's a picture of an area that a few years ago it was overgrown with blackberries, and we burned it. And so we burned it in the early spring, and by summer the wetland kind of came back itself. We did some riparian planting, but a lot of the wetland species came back on their own. The different dogwood, there's the dogwood and the tules, and cattails came back on their own. I had nothing to do with it, but it just shows, you know, that it can restore itself. I mean, really what I did was just burned this area, and the wetland came back, and I just continued to keep the blackberries out and other invasive species. But this is a really nice place. Yes?
We got 5 minutes remaining.
Okay.
Thank you.
And so talking about restoration, in different areas and wherever you're working. Traditional ecological knowledge is really good to incorporate, if possible, with the local tribe. If it's Yakima or another tribe, we really hold the knowledge, and I would say our elders hold, more knowledge than myself about the area, how it's supposed to look, how it's supposed to function, and what species are supposed to be in the area. And briefly, I like to talk about threats to the native first foods. The first one is native plant eradication, because you got a lot of, sometimes you have people moving into the county or the area and don't know what's really native, and then they're spraying and kind of eradicating. And I would say the best thing for that would be education, to going online. There's like Washington, Oregon has their native websites of native plants. Another thing that's coming up right now is the green energy projects. There's a water pump storage project near John Day Dam. A bunch of wind and solar projects are coming up with in Oregon and Washington. Also urbanization and parcelization of large properties. Here's a picture of a solar project, and also on the left is grapevines, vineyards coming up on Washington side. They're coming up all over. Another aspect I wanted to talk briefly about was climate change. The Yakima Nation has a climate adaptation plan, and it addresses a lot of the natural resources, but also it addresses like health and community. But it's a really good plan that we can look back to, look and read up on, to try to work and protect our first foods. And I like to bring this up because it's more recent, since the 2013, I think, 14, is this invasive fruit fly. It's the spotted wing Drosophila, and they're really impacting our huckleberries now. They're invasive, and growing up, I've never witnessed having this fruit fly with all the different maggots, but it's really getting bad, and hopefully we'll get a handle, or we can get some research done on it and its potential impacts of moving north, because right now it's in the southern part of the Gifford Pinchot National Forest. But huckleberries are really one of our main traditional foods that we care about. And also, like I mentioned, sharing our traditional knowledge with our children. It's important for us to take them fishing, take them hunting, take them berry picking and camping, and just sharing this knowledge with them, so that our culture will continue on. And I do a lot of education and outreach for fisheries. I do for first foods like this, and just to get the information out there about the importance of our foods to the Yakimas and the different tribes in the area, and also teaching the youth. And the youth have been really fun to work with over the years. If my presentation is available later, here are 2 resources to get additional information about native foods in the area. The Native Knowledge 360, there's a website, and also the Salilo curriculum I would recommend. And I've worked with some teachers with these curriculums, and I thought they're really useful. These are just photos of a Yakima lady with her children in the huckleberry fields. And I wanted to talk briefly about some foods that were lost and are getting rare, and that is the bridge-lip suckers. We're working on that and with the Yakima tribal fisheries to get an understanding of the current status of the suckers in the different watersheds, and hopefully we can improve habitat for them, as well as salmon and steelhead and other species. And then the wapato, and there's a link here. I'm not sure I'll have time to share with you all this, the bringing back of wapato. It's an Indian potato, and if you get a chance, maybe you can it's a really short video, but it's a really good video to learn a little bit about the restoration of Wapato in Toppenish, Washington on the Yakima Reservation. So that's the end of my presentation, and I'm thankful to be able to present to you all today about the first foods.
Great, thanks, Elaine. Yeah, I think Courtney said she would share your video in the chat if possible.
Okay.
And the presentation will be available. We'll be sending those out once we collate everything with the survey and such. So that'll go out too. So I think everyone will have opportunity to look at those videos. And I'll ask you a few questions. We have about 3 minutes for questions. The first one being, are there programs, and you touched on this, but maybe you have some more to add. Are there programs occurring with the purpose of attempting to increase the number and improve the conditions of traditional plant harvesting areas?
On the reservation, we're working to improve some of the gathering sites that were impacted maybe by logging or grazing, but mostly on reservation. And I'm interested in looking outside of the reservation because we gather a lot of different foods on state lands and federal lands, but also on the different Pinchot National Forest. We're working with them to identify areas for huckleberry restoration where they're doing a little bit of logging and burning. So yeah, we're on that track right now.
Cool. Someone asked about your project where you burned the wetland. They asked, after burning the wetland for the blackberry treatment, did you have to do anything else to prevent and treat annual grasses?
I did. We used some organic mixture of Dawn and vinegar, and we spray that on the ground and where we see some weeds coming up. But we also do a lot of hand removal weeds every year just to keep the blackberries and other invasive species out. But it's, you know, right now it's pretty good, in pretty good shape.
Cool. Look, we got several more. We got time for one more. I'm gonna look through. Someone asked, what can non-native people do to support native people's access to traditional foods?
That's one that I've been trying to figure out how to tackle. Just getting on private land to access some of our foods is difficult, and some of the tribal members don't know how to go online and look at a GIS map to find a landowner. But I've done a presentation a few months ago, and you can share my email and phone number, and if people wanted to talk to me more about the subject, and I can talk to them also. But
Okay.
yeah, that's one big awareness that I wanted to get out is, our lack of access to different places where we like to gather. Yeah.
Okay, great. Well, it looks like someone's sharing the link to the video. We're out of time right now, but we're going to share your email in the chat so people can reach out to you with questions or anything else they want to speak with you about. And thank you very much for presenting. I'm going to pass it off to Troy.
Okay, thanks. Thank you all.
Thanks, Elaine. That the purely intentional way of life and that reverence that you talk about for the species and the processes that take place out there is just really impactful. So I appreciate you sharing that perspective with us all. We are getting close to the end, and before I introduce our final speaker, I wanted to acknowledge our backup speaker, Dr. Theresa Thom. Teresa's been standing by in case we had some technical difficulties or anything like that. And so I wanted to acknowledge that and thank Theresa. She is the Aquatic Invasive Species Coordinator for the Pacific region with U.S. Fish and Wildlife Service, and did have a presentation prepared for you all today in case she was called upon. So just want to say we really appreciate that, Theresa.
All right, so as you all probably saw, Blaine's having some technical issues, so we actually are going to call in our backup speaker, Theresa. Theresa, if you could hear us, are you willing to present for the group for the final presentation?
Okay. Well, thank you, Theresa. It turns out it's a good thing we had you on deck, and I appreciate you being here. Like I said, U.S. Fish and Wildlife Service Aquatic Invasive Species Coordinator for the Pacific region. This is going to be an aquatic invasive species summary, and Theresa currently focuses on AIS-related activities throughout the Pacific region, Washington, Oregon, Idaho, parts of Montana, Hawaii, and other Pacific islands through the service's Fish and Aquatic Conservation Program. Shout out University of Georgia for the PhD, and a lot of experience from Theresa, working on water resource issues in North and South America for over 20 years. So thanks for being here, Theresa. It's all you.
And so at least I can give you some aquatic invasive species summaries. I know, Blaine was going to highlight the connection between flowering rush and northern pike, and I can speak to that as well. But I do have some more regional information that I can share. And so anyway, I am with the U.S. Fish and Wildlife Service. I cover our Pacific Northwest region. So that includes Washington, Oregon, Idaho, Montana, a little bit of Montana, and then also Hawaii and the Pacific Islands. So it's really broad and diverse for both aquatic, marine, estuarine, invasive species. And just to highlight some of the things that we are working on, especially with the from the U.S. Fish and Wildlife Service. Oops, there we go. Our region within the service, I wanted to mention that we have a invasive species prevention policy. And so we have this in place so that as field biologists, and when we're going out in the field and doing work, and not just for aquatics, this is including our terrestrial work, any activities that we're doing across the landscape, we want to make sure that we're not moving around invasive species as well. And so I just wanted to highlight some of the work that we're doing internally within our agency to make sure we're not part of the problem. Also, just to try to enhance rapid response for the potential for invasive species detections like invasive mussels, of dracinid mussels, the zebra and quagga mussels that were mentioned earlier in the quiz. We are trying to highlight work with not just responding to the invasive species, but protecting our endangered species with making sure we do consultation whenever we would do some type of a rapid response to invasive mussels. And we're working a lot with coordinating across multiple agencies, not just the U.S. Fish and Wildlife Service, but Army Corps of Engineers, the Forest Service, so that we have these memorandums of understanding agreements in place so that we can quickly coordinate and work together and share resources to respond to things. And we practice this. That's the EDRR exercise. It's the early detection rapid response exercises. We practice what it would be like in the event that we would find aquatic invasive species like zebra or quagga mussels. Who would take the lead? Who does the press releases? Who closes the area? Where do we get boat washing equipment? Those types of things, and we practice it, and we do that on an annual basis. The Department of Interior just released in January an invasive species strategic plan, and we're looking at how the different bureaus like the U.S. Fish and Wildlife Service are going to implement that. I also wanted to just briefly highlight this. We're doing some work on horizon scanning. So one of the previous talks looked at invasive species, invasions, and expansion as a result of climate change. There are some ongoing efforts to even just look at what is potentially coming to us from other countries, donor regions, that we don't even, it's not even on our radar yet. So we're scanning the horizon for new invasive species, and there's a climate change component on that. And that's done at both national and regional scales. So that's ongoing research that's, we're kind of copying what's been done in the Great Lakes And we have a project right now in the Pacific Northwest. We hope to have the results of that. Maybe at a future conference, I can present results from that, that work. Briefly, the horizon scanning, I just mentioned that, but again, putting it into the context of the bigger picture, the USGS, the U.S. Fish and Wildlife Service, also University of Florida, and Texas Parks and Wildlife. These are some of the projects going on right now, looking at what's coming into the United States and from different pathways. So organisms in trade is a pathway that's been targeted, as well as looking at boating and just water movement. So anyway, just a quick summary. And then more specifically, looking at some of these different aquatic invasive species that we're concerned about. And this is not just the U.S. Fish and Wildlife Service. There are many agencies that are working on northern pike. In fact, many of our tribal partners have passed resolutions to address northern pike as an invasive species and prevent northern pike from impacting salmon and lamprey and other very culturally important species, as we just heard. And so northern pike, if you look on the top right here, you can see from 2018, these are maps, the maps showing the points, the red dots are where northern pike have been captured. And most in the Columbia Basin, most of this is above Grand Coulee Dam. There's a lot of efforts going on in Lake Roosevelt and some of the smaller tributaries where northern pike have been found. And again, this is many groups of people, huge efforts with netting and capturing fish, also looking at northern pike as their some pretty interesting work on telemetry. So adding for live fish, putting in tags on the northern pike to see where they're moving and their seasonal use of different habitats, spawning habitat, nursery habitat, and trying to target the seasons and the habitat that they're utilizing to better eradicate and remove northern pike. This is very serious. There's there's research going on to target northern pike, and there's also kind of some unique programs to involve the public with helping this work, like reward programs. So there are a few locations where if you catch a northern pike, you alert the state to this, you can put it in the freezer, and then you get a reward back for returning northern pike or taking northern pike out of the system. It's difficult too because northern pike are native to North America. In the Midwest, especially Minnesota and Iowa, they're a native species, and it's just that they're invasive in our waters here in the West and could definitely eat a lot of salmon. They are predatory fish, especially for our native juvenile fishes of many species. And they are thought to be connected to some of the altered habitat for flowering rush. And so flowering rush is another invasive species. It's actually got a beautiful flower, and the way that it gets established and how it does establish it affects the hydrology, and so you get these more stagnant waters. Sometimes you'll see the vegetation is actually available in the winter, and so that makes great spawning and rearing habitat for northern pike, because typically we wouldn't see a lot of emergent vegetation in the winter in some of these, in a flowing river system. The difficulty with flowering rush is, trying to remove flowering rush. If you can't pull it out completely, rhizomes that are broken off can spread. The rhizomes, the roots, the little tiny pieces will spread a new plant. And so there is risk of where they are established of just downstream flow of the seeds. So really looking at source populations and being strategic on removing flowering rush. There is a cooperative weed management area that has been established for the entire Columbia River Basin, focused on flowering rush. There is a relatively new management plan that has been developed to address the impacts and the issue of flowering rush and with removal. I'm sure actually probably many of you on this call today, this webinar, probably are involved. And so again, this is an ongoing effort and definitely an invasive species that we're worried about and trying to address. And as was mentioned, I wanted to highlight a little bit about quagga and zebra mussels. They are two different species. A lot of times they're thought of as kind of evil cousins. But yeah, quagga mussels and zebra mussels originally brought into the Great Lakes in the 1980s from ballast water. And as people have moved around, we've brought quagga and zebra mussels with us. The adults they attach to submerged anything, logs, woody debris, boats, trailers, docks. So the adults have byssal threads, really fine threads, and they attach to all kinds of things. But the larvae also, they're called veligers, and they are in the water column sometimes year-round, depending on the habit or the temperature of the water and the ability for the adults to go through their reproductive life cycle. And so the concern for moving around invasive mussels is not just the structures, what they may be attached to, but also raw water. And we'll see so just to highlight, we do not have zebra or quagga mussels in the Columbia River Basin, and there's a huge effort across the West, and it's been ongoing for a long time to really try to prevent invasive mussels. There are quagga mussels in, the drainages near in the Colorado, the lower Colorado River, so that this is an ongoing effort, and constant vigilance, I can say, is what is happening related to zebra and quagga mussels. And just to showcase this, hopefully you can see this animation as zebra mussels and then quagga mussels are marching across the United States. And again, there's been a lot of coordination with many, many agencies and groups and citizen scientists and communities to, do important research on how to eradicate zebra and quagga mussels, not just an ecological impact, but also an economic impact to our infrastructure as well. Hydrilla, again, this is another aquatic plant that we're looking at and concerned about. And a difficulty with hydrilla is that it looks a lot like some of our native water plants. So American waterweed is native. And again, this, I don't know if you've heard of, for identifying poison ivy, leaves of 3, let it be. That kind of works for Alodia as well. Our native has 3 whorls on the leaves. And so let it be if you see the 3. If there's more than that, it's potentially one of our invasive Brazilian waterweed or hydrilla. There you go, there's a little arrow. Crayfish, that's been mentioned as well. We do have our native signal crayfish. I got this one right on the quiz. the rusty crayfish, you can see on the right, where you would grab the crayfish to try not to let it pinch you. That's where it's got these kind of a rusty thumbprint, and it looks like a rusty spot, and that's a great diagnostic feature on how to identify a rusty crayfish. Again, one of the differences with trying to monitor or detect crayfish And this is where, the eDNA piece is really interesting, trying to help monitor for these species. But, nighttime surveys, crayfish are potentially more active at night, and it's pretty easy to see them in the streams with their eye shine if you have a headlamp. But rusty crayfish, when they do become established, their population numbers are intense. They outcompete the natives, and they really are ecosystem engineers. They change the streams where they're found. And then just some other invasive crayfish that we're on the lookout for: red swamp crayfish, and then the ringed crayfish. Again, one of the diagnostic features there are the dark bands on the tips of the claws. Again, highlighting ways to report our aquatic invasive species, definitely through the USGS Nuisance Aquatic Species Database. That is a great tool, and it's been enhanced. If you haven't used it lately, you can now, if you're planning to do site visits, you can actually do a geospatial search. So you can go onto the website and take their little tool that they have, draw a circle around your field sites, and you can look at all the different reports of aquatic invasive species in that area. So that's an interesting feature. There's a lot of new features on there as well, with flood tracking. So if there's flooding that happens, it can help you predict where you might want to do additional monitoring post-flooding to see if maybe invasive species have spread as a result of the high water. EdMaps, talked a little bit about monitoring. I'm sure many of you use EdMaps. In fact, there's a webinar coming up at the end of March through the North American Invasive Species Management Association, highlighting EdMaps. It's the March 31st and April 1st, with all kinds of different tools and features on utilizing EdMaps to the best of your ability, new tools and tips and how to import large data sets. And then I'm going to just give a plug to our state invasive species councils as well. All of our invasive species councils in this region are very active and involved in all kinds of invasive species work, terrestrial and aquatic, and also outreach to everyone on helping report invasive species. And then creating lists and ways to control things and coordinating. So they're really fantastic resources. And again, just, we all have our ability to do what we can to stop the spread of aquatic invasive species. We have watercraft check stations. There's all other types of campaigns, Don't Let It Loose, really focused on pet releases and classroom releases. Don't spread invasive species, even with the terrestrial campaigns of Play, Clean, Go. So just a lot of our outreach campaigns really trying to target clear messaging and our different user groups, including us. And we do our best to make sure our gear is clean, drain, and dry. And here's my contact information, and I'm happy to highlight any of the work that we're doing here. I'm glad to share our prevention policy. I'm glad to talk with you about any of our activities that the U.S. Fish and Wildlife Service is doing. And if you have any other questions, and I apologize, I feel so bad that Blaine was unable to speak to you, but I know that the story between flowering rush and northern pike is really, really interesting. And, it's a concern that, especially for northern pike, if we're not addressing the flowering rush, that's habitat that they can find and move downstream. So anyway, thanks. I think we have a little time for questions.
We do. Thanks for hopping in there, Theresa. I really appreciate it. I don't see any questions sent in here. So if anybody has them, someone said, Not related to you, but would Blaine be able to send his presentation to the attendees? I believe that is going to be the case. It will be included. Any other questions for Theresa? There might be something else, but I can't read it. So with that, I'm going to pass it off to Troy. Thank you so much, Theresa, especially for hopping in last
No problem.
Yeah, we appreciate that, Theresa. And if you have any more of those coats laying around, that coat you're wearing at the end is super sweet. I'd love to have one of those. So yeah, we'll do our best to get Blaine's presentation uploaded for folks, and maybe there's an opportunity in the near future to set up a short webinar that's targeted for that type of stuff. I know there's some moving and shaking with the Flowering Rush Working Group right now, and so there may be some opportunities as we move forward to dig into that a little bit deeper. One thing I want to mention as we close this out is there is going to be a survey that is activated as we close out this webinar as you leave. Please, if you have the extra few minutes, do fill out that survey for us. We'd really appreciate it. Thanks to all of the presenters for your great presentations and your hard work getting prepared for this. We appreciate your time. And at this time, anybody here who is seeking recertification credits through Oregon or Washington, now is the time to enter your ODA or Washington Department of Ag license number in the chat box. And so if you have a group that's attending on one computer, then please have all the names and have everyone send it in, whether it's one big chat or you each hop on there. Let's make sure we get every person's license number in there, and we'll match those up to the registrations and get those submitted to their respective Department of Agriculture. So with that, we're gonna close this out. So I don't know if there's any more last words from any of the other organizers. Great job putting this together, guys. Really good job. So, and thanks again to everyone who's here. Have a great weekend.